with the term Improving Collaborative Research between Netherlands, Japan, and Indonesia. This symposium will offer learning and networking opportunities through multidisciplinary invited speakers from Indonesia, Japan, and also Netherlands. Professor Dante Saxono, as the Vice Minister of Health, Republic of Indonesia, will become the keynote speaker in this symposium. And interestingly, the other keynote speakers, Prof. Patrick Rinson from Leiden University and Dr. Ippei Shimizu from Jutendo University, Japan, will share a different perspective on the role of brown adipose tissue in the link of metabolic, cardiovascular, and aging. This symposium generally will be divided into two main sessions. The first session is the article and publication sessions that include the meet editor from Nature Metabolism and Frontiers Journal, and meet the professor session to give a good perspective on how to publish in a high-impact journal. And also the young scientist sessions for the fresh graduated student for determining the best career option for them. And the second one is the scientific session that will provide various high-qualified speakers who will talk about regeneration therapy, genetics, herbal medicine, big data, and some clinical issues that related with metabolic, cardiovascular, and aging area. Last but not least, welcome to our international symposium. Enjoy the international conference and please add your voice to the discussion. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Jeffy, for your speech. Next, we would like to invite the head of MVA cluster in IMERI, Dr. Diki Levedus Tapari, to give his speech. To Dr. Diki, the time and place is yours.
Okay, thank you, Natasha. Good afternoon to all the attendees. Also, maybe good morning for all those attended from Europe area. First of all, I would like to welcome all the attendees as well as honorable guests, and of course to Professor Badrul Hegar as the head of the Indonesian Medical Education and Research Institute. I would like to thank you very much for your kind support, and also for Dr. Jeffrey, of course, for the one who started to prepare this event starting from 1 year ago, and he's striving to invite all the best available speakers, especially from Netherlands, Japan, also Indonesia, to share their expertise on the aspect of cardiovascular, metabolic, and aging. So from this symposium, we hope in the future we can further collaborate in terms of research as well as education or other aspects. Hopefully this sharing experience between many experts from 3 different countries could foster further collaborations. So not only we will learn about the updates on the scientific knowledge in the aspect of cardiovascular, metabolic, and aging, as mentioned by Dr. Jeffy, but I think the symposium gives a unique perspective as well, because we also will share with editors from various well-known journals about their perspective on how to publish articles in their journals. And we also have another sessions in terms of how to become a scientist in Indonesia. We know that becoming a scientist in Indonesia, we have many problems, but hopefully with a session of sharing experience by well-known experts and scientists, we can all learn how to develop ourselves as a scientist. So thank you once again for Dr. Jeffy for organizing this very interesting first symposium on cardiovascular, metabolic, and aging. And hopefully by this symposium, not only we will learn about the latest scientific evidence, but also we will foster further collaboration with all the collaborators. And we'd like to invite as well for all the attendees, if you wish to collaborate with the IMERI, especially from the MVA cluster, you are most welcome to contact us. Okay, without further ado, I think that's all for me, and I would like to give the time back to Natasha. Thank you.
Thank you, Dr. Diki, for your opening remark. Next, we would like to invite Professor Badrul Hegar as the Director for Indonesian Medical Education and Research Institute, University of Indonesia, to give his opening remarks and open the first CMARS 2023. To Professor Badrul Hegar, the floor is yours.
Okay, thank you very much. Good afternoon all. The Honorable Head of Metabolic Disorder, Cardiovascular and Aging Research Center, IMERI FKUI, Dr. Diki Levenus Tahapari, PhD, SPPD, KMD. Also the chair of the first CMARS, Dr. Agian Jevillano Barindra, PhD, and of course, all the participants. First of all, I would like to congratulate to MVA Research Center and also the community for holding this symposium. It is a challenging initiative to invite us all to look the problem of metabolic, cardiovascular, and aging, which we are now very complex from a convergence perspective. Therefore, I would like to express my deepest gratitude to all the experts from the Netherlands, from Japan, and also from Indonesia, who have spent their time to share knowledge and experience on this symposium. I hope this initiative will become a vehicle to complement the existing networking and collaboration in discovering novel ways to diagnose, treat, and prevent diseases in metabolic, cardiovascular, and aging, and also novel technologies enhance the research for future health professionals and scientists, and also bring innovation from the laboratory benches to the clinic and the marketplace, or vice versa. We know that no scientist in the world has all the talent and capability to solve today's problem. More people can do better than if they work independently of each other. So why we need networking as about building authentic and trust and relationship and seeing how we can help others. Academic networking itself can play a crucial role in young researchers' careers, both on social and professional level. So why we need also collaboration, that we can find someone who need us just as much as we need them, benefit both parties equally. Once, of course, IMERI is greatly assisted by this initiative in creating better healthcare and quality of life through disruptive innovation, especially in developing convergent research in metabolic, cardiovascular, and aging. I hope through this initiative, a thematic focus of research will emerge with problem-based approach to complex issues in medical science, health, and disease, not only relevant to global perspective, but also to its country community. There will be also a talented researcher who are mature under inspiring leadership. Multidisciplinary scientists and center of excellent collaboration need space and support to address their complex scientist challenges. Thus, once again, I want to thank you very much to all very much for the initiative, and I hope this is a step further in building and sustaining our research capacity. Okay, thank you very much. Now, officially, I open the first CMARS 2023. Thank you very much and enjoy the symposium.
Thank you very much, Professor Badrul Hegar, for your opening speech and to open our first International Conference of Cardiovascular, Metabolic, and Aging Research Symposium 2023. To start our symposium today will be a keynote lecture titled Brown Adipose Tissue as a Therapeutic Target for Age-Related Diseases by our speaker, Dr. Ipe Shimizu. This session will be moderated by Dr. Renan Sukmawan. So without further ado, We would like to welcome our moderator. Good afternoon, Dr. Renan.
Good afternoon. Very warm. Good afternoon, my colleagues, ladies and gentlemen. Okay, can I start, or you want to say something?
Well, we're going to start with a photo session.
Okay, offer the session. All right.
Before we start our lecture, we want to invite every single participant here to turn on your camera. So we will have a photo session together. We will wait for 1 minute until 13:24 to wait for everyone to open your cameras. Thank you very much for opening your cameras. While we wait, I will give the floor to the documentation team to take the photo. Very well. I will help in doing documentation. So everyone could smile and put on your best smile. I'll count to 3. 1, 2, 3. Once again, 1, 2, 3. Very well. Thank you very much for turning on your cameras. Now, before we continue to the next session, allow me to introduce our moderator for this keynote lecture, Dr. Renan Sukmawan, by reading his CV. Dr. Renan Sukmawan is from the Department of Cardiovascular, University of Indonesia. He has conducted a doctoral degree in University of Indonesia, also his PhD in Kawasaki Medical School, and studied cardiology in University of Indonesia. His activities composed of being the head of the Department of Cardiology, Vascular Medicine in Faculty of Medicine, University of Indonesia, and a lecturer in Department of Cardiology, Vascular Medicine, and Faculty of Medicine of University of Indonesia. Very well, after we've come to know our moderator, I will now pass the spotlight to our moderator. So to Dr. Renan, the floor is yours.
All right. Thank you, Natasha. Assalamualaikum warahmatullahi wabarakatuh. Very warm good afternoon, my colleagues, ladies and gentlemen. It's a pleasure for me to host this session. We will hear an update, the topics that has been one of intriguing, I would say, intriguing issues in cardiovascular medicine and metabolic. I think we already know there are wide adipose tissue, there are broad adipose tissues, and recent studies has shown that if you have the presence of broad adipose tissue, it's correlated with lower incidence of cardiovascular disease, better glucose metabolism, and some beneficial things for our health. Now we will hear more what will happen on this and how this will increase our understanding and again will incorporate our practice in the future. I would like to invite our guest speaker for this first session, Professor Shimizu. Natasha, can you help us with the CV, please? Right, Professor Ipe Shimizu from Department of Cardiovascular and Medicine at Juntendo University in Japan. Currently, he's associate professor at Department of Cardiovascular and Medicine at Juntendo. He has lots of publication, a member of lots of respective scientific associations. As we can read here, he's also member of ISHR, Mid-Care Investigator, and also member of GST Forest Program as a lots of publication by seeing this CV scleris has a tremendous record in international publication. As we saw here, Professor Shimizu also editorial board member of a lot of journal here, scientific journal molecular and cellular cardiology, and also as a fellow of European Society Cardiology and also American Heart Association. Without further ado, please, Professor Pei Shimizu. Konnichiwa, Professor Shimizu. Yoroshiku onegaishimasu.
Thank you very much.
To say something in Japanese. Okay, please, Professor. We will hear for your. We'll discuss after your lecture, please.
Oh, thank you very much, Professor Ren and Squamman, for your kind introduction. And I would also like to thank the Jeffy and the organizer for inviting me to this great symposium. So I hope you see what I see now. Is it? Oops. All right. So I'm Ipei Shimizu from Juntendo University in Tokyo, Japan. Thanks again for this kind invitation. So our group, I'm a cardiologist, but our group has so far extensively studied the role of aging in the adipose tissue or endothelial cell in the context of heart failure, obesity, and diabetes. Today, I would like to make a deep dive in the brown adipose tissue dysfunction in the pathogenic role of the dysfunction in brown adipose in the heart failure and obesity and diabetes and some other disease.
Your presentation slide cannot see.
Yeah, it's like blank or blank presentation here.
Really?
Okay.
How about this?
Okay, yeah.
How about this?
Not yet.
Not yet, really.
Once again, probably you have to re-share.
All right, I will share my desktop and hope. How about this time?
Yeah, I think it's clear. I think everybody sees clearly.
Yes, we can see.
Oh, you can see. All right.
But not in the full screen setting.
Yes.
All right, all
Okay.
right. I'll see. How about share this and then it is a non-full screen.
Not yet.
How about this?
Yeah, I think it's already full screen. Please.
Okay, all right. Thank you very much. I apologize for that. So today I would like to make a deep dive into the brown adipose tissue. We have pretty much interest in aging process, heart failure, obesity, and diabetes, but I will skip these. All right, so as we know, the obese population is rising. And then the keyword will be how we can make kind of a systemic metabolic health. Also, one of the approach will be to make a kind of rejuvenation for the white adipose tissue, the rebrowning of the brown adipose tissue, or beiging of the white adipose tissue. The point is that when we make a maintenance of homeostasis for these adipose tissue, they contribute for the systemic metabolic health, and then suppress pathogenesis in obesity, diabetes, heart failure, and unfavorable aspects of the aging. So as we know, we have to make it very simple, we have two types of adipose tissue. One will be described as brown, another will be described as white adipose tissue. The distribution of these adipose tissue are different in our bodies. So this is a study from our data from mice. So brown looks brown, and then white looks white. And then when we impose them in the high-fat and high sucrose diet, they increase in the volume, as you can see here. But it's clear it's a kind of a different organs, as you can see here. And in the high magnification, the brown adipocyte looks quite different from white adipocyte. They're really enriched in mitochondria. And then initially, it was characterized with the cell contributing for the heat generation. And then brown adipose tissue, as you know, is highlighted because they are very active metabolic organs. And in humans, still there are controversies exist, I know, but the 63 grams of brown adipose tissue are predicted to be present in humans. And then continuous activation is reported to lead to the 4.1 kilogram weight loss per year. So this is the data from mice. It's the ratio for the tissue weight is as small as like 5%, but still when they get the highly activated, the contribution for the triglyceride clearance and also the glucose disposal is almost like 50 to 75%, at least in mice. It's a kind of enormous active metabolically active organ. So in humans, the brown adipose tissue is known to exist, but mainly in newborns and small rodents. As you know, going back to 2009, these signals for the PET-CT scan were considered to be a kind of non-specific signaling, but it was known that the black signal matches with the presence of brown adipose tissue, and then the activity of the brown adipose tissue decline with aging and an increase in body mass index. So related with the TV show, I apologize, some of them were in Japanese language. I don't know if you know this person. So his name is Wim Hof, Mr. Wim Hof, and he's described as the Iceman. Then, so this person has a kind of a clue how we can activate brown adipose tissue. It's kind of an amazing guy who can even swim in the icy water. So never do this. The point is that the cold exposure activates brown adipose tissue. And then this paper in the PROS one extensively studied this person. And then Iceman has a twin brother. So he's a kind of an ordinary man. Iceman likes the cold exposure. But the point is that both of them has a very active form of brown adipose tissue. So their age, the chronological age was around 55, I remember, but the activity of the brown adipose tissue was almost like in the people who aged like 30 years old. So it's kind of a chicken or egg question, but the cold exposure, it will be one approach to activate this metabolic organ. Then interestingly, like in the type 2 diabetes patients, dyslipidemia patients, coronary artery disease patients, and heart failure patients, and heart hypertension patients. So these are individuals with age-associated disease has a low activity for brown adipose tissue. So again, it's kind of a chicken or egg question whether there will be this is going to be a kind of a therapeutic approach remains to be an open question to be explored. So this is Ken Walsh. I was a postdoc and in the second year as an instructor of medicine in Boston University. So what we did was maintain mice on high-fat diet, and then focus on interscapular brown adipose tissue, like capillarization. So the green signal is the capillary. So as you can see here, the high-fat, high sucrose diet diminished capillarization in the brown adipose tissue, in the interscapular brown adipose tissue. And then this associated with a whitened phenotype in this organ. And what we found was that the vascular endothelial growth factor A, the angiogenic form, VEGF, was reduced on the dietary obesity, and this increased hypoxia-inducible factor 1A, HIF1A. The HIF1A is not an angiogenic molecule in the adipose tissue. And then instead, they promote autophagy, and then leads to the clearance of the mitochondria, and then shows a kind of a whitened phenotype. And this leads to the systemic insulin resistance. And then when we re-derivative VEGF-A in the obese brown adipose tissue, they show again the re-brown phenotype. Right, so metabolic stress, fatty acid promotes whitened phenotype in the brown adipose tissue. After returning to Japan, I continuously I'm also studying the brown adipose tissue. So what will be other factors? Like using DNA microarray, proteomics, and metabolic analysis, and then analyzing these, integrating this information with the bioinformatics. So what we did for the metabolomic study in the brown adipose tissue, we found that in the high-fat diet mouse, we analyzed neurotransmitters in the brown adipose tissue and got interested in the evidence that the gamma aminobutyric acid, GABA, was only the only one neurotransmitter that showed an increase in the brown adipose tissue. The others like norepinephrine showed the reduction, serotonin reduction. Some of them are not detected. Initially, I thought that the increase in GABA should be protected, but it was quite opposite, interestingly. So GABA binds to the GABA B receptor 1. So we generated, we analyzed the GABA B receptor 1 systemic knockout mouse on the high-fat diet, that they show the browning phenotype. And this is a schema of the finding. The hypoxic HIF1 alpha axis contributes for the translocation of the GABA B receptor 1 into the cell membrane, and then together with the ligand GABA, the GABA B receptor 1 axis contribute for the influx of calcium into the mitochondria through the MCU transporter, and then the excessive calcium contribute for the reactive oxygen species production, and excessive loss leads to mitochondrial dysfunction, and then brown adipose tissue dysfunction. All right, so I would like to talk a little bit about the coagulation factors. So as you know, the coagulation factor 10A, fibrinogen, fibrin clot, blood clot axis. This is a kind of a common pathway for the coagulation cascade. But we found that the coagulation factors also is produced locally in the brown adipose tissue and the brown adipocytes, and then also the excessive signal pathway from coagulation factor promoted brown adipose tissue dysfunction. So we analyzed the public database in the obese mice in the brown adipose tissue, and got interesting evidence that the coagulation factor 7, factor 10, and the receptor like protease activity receptor 1 PAR1 was increased in the brown adipose tissue. And then we analyzed in our data transcript study show that the factor 10 was increased, and activated form factor 10A was increased in the brown adipose tissue on the high-fat diet, as you can see here. And also the high-fat diet leads to the whitening phenotype in brown adipose tissue. And we also found that tissue factor was increased in the ELISA and also the immunofluorescence study. So we also got interested in the evidence that the factor 7 was increased in the brown adipose tissue in transcript and also in the protein level as analyzed with immunofluorescence. So this is the summary of the coagulation cascade. Factor 7 gets activated from factor 7A, and the tissue factor increase and also factor 7 increase, factor 10 increase in transcript, and also factor 10A increase in the brown adipose tissue. So this is the complex of factor 7A, 10A, TF, tissue factor. It's not pretty much focused, but they are also important for the mediating the signaling pathway, like PAR, protease activated receptor 1 and 2 pathway will be activated through these coagulation molecules. So another group reported a PAR2 signaling pathway in visceral fat promotes insulin resistance and then have a pathogenic growth of obesity. But the role of PAR signaling in brown adipose tissue was not pretty much highlighted. So we've got interested in the evidence that in the brown adipose tissue, they don't have many PAR2. So white adipose tissue is predominantly expressed PAR2, but as you can see here, there seems to have a kind of a minor role in brown adipose tissue. But in contrast, PAR1 was predominantly expressed in the brown adipose tissue, and then with a high-fat diet, PAR1 increase, as you can see here. So these are Western blot data, and also for the staining data showing that the brown adipocytes seems to be the predominant source of the production of PAL1. So we tested the role of factor 10A inhibitor rivaroxaban, and then on the administration of factor 10 inhibitor are promoted, the partially rescued the whitened phenotype of the brown adipocytes, as you can see here. Also in the dietary obese mice, when they are post-oral exposure, the thermogenic response will be reduced, as you can see here. This phenotype was improved with administration of factor 10A inhibitor, as you can see here. And then systemic glucose intolerance, as you can see here, in the hyperdiet mouse were improved with administration of factor 10 inhibitor. So the suppression of factor 10 PAL1 signaling pathway seems to activate the brown adipocyte function, as you can see here. And also, we analyze the factor 10A inhibitor, the level of reactive oxygen species with DHE staining, the dehydrolysis regime staining show the reduced signal with the delivery of factor 10A. So the level of loss seems to be reduced in the obese brown adipocytes, as you can see here. And then we also analyze the oxygen consumption ratio, and then as you can see here, the VO2 was increased and VCO2 increased, and energy expenditure increased with our administration of factor 10 inhibitor. And also the electron microscope findings show that in the obese mouse, the mitochondria becomes like a fragmented, and some of them show the unhealthy morphology. This phenotype was rescued with administration of factor 10 inhibitor, as you can see here. So at this time, we are mainly focused on the mitochondria in the around the capillary. What is interesting is that in the clinical setting, we prescribe warfarin, not that much compared to the previous date, but warfarin we prescribe together with a factor 10A in atrial fibrillation patients. So it's kind of a anticoagulation drug, as we know, but biologically it's clearly different from the factor 10 inhibitor. In the hyperdiet mouse, when we prescribe warfarin, the whitened phenotype of the brown post tissue doesn't improve at all. And also level of the reactive oxygen species remains high, as you can see here. So there's no difference between control and warfarin treated mice on the hyperdiet. Also the core tolerance test, glucose tolerance test, and also the energy expenditure of the mice doesn't change with administration of warfarin, as you can see here. So warfarin and factor 10 inhibitor, they are clearly different drug, as you can see from these data. So the warfarin doesn't affect the mitochondria density, as you can see here. Some of them looks really fragmented, and also the number would be diminished, as you can see from the compared with the control group. So it's quite similar. And then we analyze the tailbone breathing time, thrombin activity, level of factor 10A. So as you can see here, the warfarin factor 10 group, both of them have a similar level of the tail breathing, thrombin activity, and factor 10A. But still, as you can see, the effects, biological effect of the brown post tissue is clearly different between these two drugs. And also, we generated brown post tissue specific PAL1 knockout mouse. So UCP3 mouse was crossed with a PAL1 flox mouse. And then please take a look at the interscapular area. Well, in the obese mice, so these are different mice, pre and post, the cold exposure doesn't increase the thermogenic response in the interscapular area, as you can see here. This looks a little bit enhanced. But if you take a look at the brown post tissue specific PAL1 knockout mouse on the high-fat diet, the emission for the thermogenic response seems to be enhanced, as you can see here. So if the mouse, the PAL1 receptor in the brown adipocyte was specifically depleted, this leads to the enhanced thermogenic capacity in mice. And also we did a kind of gain of function study and administered like a tissue factor 3 and PAR1 encoding genes F2R. So this is kind of a tissue factor and PAR1 gain of function model administered through adeno-associated virus. Also, the brown adipose tissue was injected with AAV for these two molecules. And as you can see here, these mice are kept on the normal CHO. They show the whitened phenotype, and also the DHE staining was enhanced, as you can see here, showing that the enhancement of these two molecules leads to the increased reactive oxygen species in the brown adipose tissue. And also, external microscope findings suggested show that the fragmented mitochondria and also the reduction in the density of the mitochondria, as you can see here. And also in the normal fed mouse, the activation, the gain of function for tissue factor and PAR1 on the normal CHO showed the reduced thermogenic response, and also the systemic glucose intolerance was enhanced with administration of the F3 and F2 into the brown adipose tissue. So this is a summary of our findings. So on the hypoxia, again, increased HIF1 alpha under metabolic stress condition. HIF1 alpha is not an angiogenic molecule in the brown adipose tissue, as I told you, but instead, this increase the transcripts, factor 3, tissue factor, also coagulation factor 7, 10, and PAR1 in the brown adipocytes. And then like a autocrine or endocrine way, they activate like PAR1 pathway, and then again PAR1, the calcium calcium is located downstream of PAR1 signal pathway, and then again the mitochondria become overloaded with calcium and reactive oxygen species or increase, and then this leads to mitochondrial dysfunction, and the brown adipose dysfunction leads to systemic glucose intolerance. All right, so we like to change the gear, and then I would like to talk the concept of systemic glucose and metabolic cell protein. So I extensively studied the aging process, but what is interesting and mysterious, why they make a kind of a synchronization. Some of them, like brain or heart, are like a terminally differentiated organs, but instead, like bone marrow, they are highly proliferated. We share a kind of a concept that they show functional decline with aging. So the point will be that in circulation, the pro-senescent molecules like xenometabolite I define, and the xenoprotein I define in circulation may have a causal role for the synchronization of aging, so-called sync aging. All right, so I will show you why I came across this idea through the study for heart failure. All right, so heart failure is an increase like a pandemic, and 1 to 6% of the whole population is suffering from these deadly disorders. Also, like as you can see here, the kind of silent epidemic is sometimes described. It's still so many medical needs underlying this deadly disorder. So this is a study from humans when we focus on the metabolite in circulation in the heart failure patients. Some of the metabolites is reported to increase, and some of them are reported to reduce with aging in circulation. And also on the red molecules are tested in humans. Also in the failing heart, some of the metabolites were increased, and some of them are reduced in the failing heart. All right, so the heart failure is closely related with the metabolic remodeling. But the question will be, what will be the interaction between metabolically active organs, including brown, white, liver, skeletal muscle? There's so many things that are left behind to be explored, I guess. So this is an interesting Kaplan-Meier curve showing the Kaplan-Meier curve from heart failure patients. So the red group, they have a very poor prognosis, as you can see here. And the point is that these patients have a reduced body temperature in the inpatient clinic, inpatient as well as outpatient clinic, as you can see here. So if the heart failure patients, the body temperature become below 36.3, the prognosis become very bad, as you can see here, compared with other groups. So interpretation of, we can interpret this data in several ways. We try to find the underlying, to test the potential role of branch post tissue in heart failure. So I was a member of Niigata University like 2 years ago, and then first we tested the reproducibility of the previously published data from Europe. So in the heart failure patients, so most of them, I mean, all of them were dilated cardiomyopathy patients. The ejection fraction was half, and then together with left ventricular dilatation, developed in the heart failure patients. They are 10 years younger than the control group, but the body temperature became as low as around 36.0 centigrades. So even though they are younger, the body temperature was reduced in heart failure patients. So we tested generally the TAC operation, like thoracic constriction in mice. It's kind of an acute aortic stenosis model. And then the left ventricle is exposed under pressure overload. And then they show a kind of failing heart phenotype. So we tested the thermogenesis in the mice. So morphologically, I mean, the phenotype in this phase seems to be quite similar. But if you take a look through the thermogenic camera, they look quite different creatures, as you can see here. The emission of the temperature is reduced in heart failure patients, as you can see here. And the point is that whether when we make an intervention for these mice through the enhancement of like sauna condition, or when we warm them, the point will be whether they become a therapy for heart failure. So we analyze the branch post tissue on the TAC operation, and then we found that the interscarpal area, the interscarpal branch post tissue become hypoxic, as you can see here from brown on signal. And also we found that the tunnel positive cell will increase with TAC operation. Also the branch post tissue weight was reduced in TAC operated mice. Also to test the role of potentially diminished branch post tissue function, we made a TAC operation in branch post tissue removal of mouse. So if we remove surgically the branch post tissue, the body temperature was reduced compared with sham operated mice. And then we made a TAC operation in these mice. And then branch post tissue removal led to the reduction in the fractional shortening, together with a left ventricular dilatation, and also the branch post tissue removal enhanced the fibrosis in left ventricle, as you can see here from muscle trichonal staining. And we got interested in the evidence that the trimethylamine N oxide TMO increased with a branch post tissue removal. So branch post tissue removal together with TAC operation led to an increase in TMO in the plasma and also in the heart, as you can see here. So in general, so this is a very famous paper published in 2013 in Nature Medicine. So I think in general, the carnitine, choline, the choline derived from these food leads to the production of trimethylamine, TMA in gut flora, and TMA will be converted to TMO in the liver through the oxygenase 3, FMO3. So TMA promotes atherosclerosis and then leads to the heart attack, stroke, death, and revascularization. So it's kind of a bacterial derived metabolites like a TML. So in the heart failure patients, we also confirmed that TMA was increased in the plasma. Again, the TMA increased with TAC in plasma and heart in mouse. And when we took a look through the electron microscopic findings in the TAC operated mice, together with TMA introduction, it's clear that morphologically dysfunctional, unhealthy mitochondria increase with introduction of TML. And we went further test the mechanism and did some proteomic study and got interesting evidence that the cytochrome C oxidase subunit 1 in the complex 4 in mitochondria became not detectable with a proteomic study. And TML led to the reduction in COX-1 in the mitochondria from cardiac tissue. And then mechanistically, we got interesting evidence that the HEM-A and HEM-B were reduced in the TML. And then to protoporphyrin 9 and Fe2 plus become HEM-B and 2H. So fetch ferrochelatase is critically important for this reaction. And TML, we consider that binds the Fe2 plus, and then we consider that the TML suppress this metabolism by fetch. And then it's a very mild findings, but was still statistically significant. But when we introduce the testis response together with TML, protoporphyrin 9 peak area was significantly increased, suggesting that TML suppress the metabolic pathway for fetch. And then HEM-B was reduced, as you can see here. And then this leads to the unstable, diminished the stability of the COX-1, leading to reduction in the COX-1 in mitochondria. So this is our hypothesis. All right, so we are still interested in the TML in the context of sarcopenia. So TML has a mediated pathogenic role for heart, and then also the skeletal muscle is enriched in the mitochondria. So we analyze what will be the level of TML in the aging, and in the aged mice, the TML will increase, as you can see here in circulation. We generate TML infusion model, and then tested the mouse in the middle-aged mice. So the forced running distance for preliminary examination test clearly show that the TML load introduction reduce the distance, running distance, as you can see here, and also in the quadriceps, tibia, alis anterior, and the other skeletal muscle. And TML introduction led to the increase in the unhealthy morphology of the mitochondria, as you can see here. So they look like a balloony phenotype together with the crystal seems to be like a diminished or unclear. So we consider the TML contribute for the pathogenesis in the sarcopenia. So this is a study done with ongoing studies, including Yoko Yoshida and Takashi Iso and other members. So TML promotes heart pathogenesis in heart failure and also in sarcopenia. And what will be other disorders? So now we are collaborating with Tohoku University, Professor Koshiba's group, and analyze more than 1,000 individuals altogether. And got interesting evidence that these healthy individuals has, together with in parallel with aging, the TML level increase in circulation, or even in the young population, some of them has a very high expression profile for TML. So I think they have a poor prognosis for these individuals because TML, at least in mice, destroys mitochondria. So maybe these young population are prone to develop heart failure or sarcopenia compared with other rather lower individuals showing the lower expression profile for TML. So other groups show that the TML level was increased in the cerebrospinal fluid in the Alzheimer's disease patients. And also some group tested the TML introduction model in young mice, and then the cognitive function seemed to be reduced with the TML introduction. Also, another group tested the level of TML in the Parkinson's disease patients So in the cerebrospinal fluid, in the Parkinson disease patients show the increased level of TMAO in the CSF and also in the plasma. So as you can see here. So what I'm trying to do with the consortium member is that I'm like trying to make a kind of a new concept of the disease, like xenometabolite related disorders, SRDs in short. So for example, at least TMAO was increased in circulation in healthy individual and also the healthy mice, and then they destroy mitochondria. So SRDs include like heart failure, sarcopenia, Alzheimer's disease, Parkinson's disease, and some other disease. The point will be the organs enriched in mitochondria will be damaged through increase in TMAO. So these will be should be a kind of a syndrome. Large clinical studies show that they overlap in many aspects. That heart failure patients are prone to Alzheimer's disease, and then Alzheimer's disease patients are prone to heart failure. Sometimes simply because they forget to take drugs for the heart failure, for example, or non-communicable disease, including diabetes or hypertension. But I think they share some pathogenesis. Some of them, I believe, will be through TMAO. So through the consortium members, I'm trying to make a drug targeting like TMAO and other metabolites. So I will a little bit go back to the brown adipose tissue and then focus a little bit on the capillary. So this is the electron microscope finding I did. The brown adipocytes are surrounded with a capillary. And then, of course, other like heart or skeletal muscle, always this kind of the cell are surrounded with a capillary. So I a little bit share about the TMAO in the capillary, and also I will talk about the protein in the capillary in the end. The capillary network formation is really interesting. And then this is a study from the kidney with the aged mice, like 6 months of age, like 18 to 22 months of age mice has a diminished capillarization, as you can see here. So this is a study from kidney in mice. Also, this is a study from the cortex, the brain, on the hippocampus in the aged mice. So compared with the young mice, aged mice has a diminished capillarization, as you can see here. Like a CD31 positive signal will diminish in the organ. And also in the cardiac tissue, compared with the young mice, like aged mice has a diminished capillary, as you can see here in the cardiac tissue. So we going back to 2010. So this is my thesis. I graduated with this paper. So when we make a TAC operation in mice, they have the vascular density was reduced with a left ventricular pressure, as you can see here. And also another group showed that in the heart failure, HFpEF, heart failure with preserved ejection fraction, HFpEF patients, the capillary density seems to be reduced compared with the control group, as you can see here. So this is also a very famous paper, I think, that published in Science in 2021 with aging, the soluble form VEGF receptor 2 was increased and then block the VEGF mediated pathway. So the VEGF enhancement even increased the lifespan in male as well as female mice. Also, the beta-gal signal increase in the 24 months of age mice, but administration of VEGF diminished this signal. So it's the enhancement of capillarization continues to be an interesting approach for the longevity. And also, we tested the level of SGLT2 inhibitor empagliflozin in mice. And then we generate TAC operated mice, and then some of the group are administered with empagliflozin, the SGLT2 inhibitors. Like humans, the reduced ejection fraction or improve its administration of SGLT2 inhibitors, empagliflozin, and also left ventricular dilatation was reduced under this setting. And if you take a look at the left ventricle, TARC led to the reduction in the capillarization in the left ventricle, but the empagliflozin administration enhances is quantitative data. So it's clear that empagliflozin mediates a cardioprotective role through the enhancement of the capillarization. So in this paper, we focus on the end-cell cell. TARC operation reduce phosphorylated enos. Empagliflozin increases. And then in parallel, nitric oxide are reduced with TARC, and they improve with empagliflozin. And also in the end-cell cell, tunnel-positive cell increase with TARC, but diminish with administration of empagliflozin. So this is a scheme of our findings. Right, so on the left ventricular pressure overload, the catecholamine increase reactive oxygen species, and then this promotes end-cell cell apoptosis, and then capillarization would reduce, that leads to left ventricular dysfunction. What EMPA does would be EMPA increase 3-hydroxybutyrate, and then this butyrate, a ketone body suppress level of reactive oxygen species, and then ROS reduction ameliorates the suppress end-cell cell apoptosis, and then contribute for maintenance of capillarization. And the AKT enos pathway, NO pathway also contributes suppression of ECI apoptosis. SGLT2 inhibitor will be one approach for the enhancement of capillarization in the heart. And also, this is Yo Furucci, a lab member, and he was interested in the polyphenol. And then this is a study from the brown adipose tissue. In the high-fat diet mouse, the capillarization was also reduced in the brown adipose tissue. And then this would improve its administration of polyphenol, boysenberry-derived polyphenol. And then again, we focus on the cell death in the endothelial cell. On a high-fat diet, increase endothelial cell death in the brown adipose tissue. And this was suppressed with administration of boysenberry polyphenol. And together with the endothelial cell specific SART-T1 knockout model, we found that the boysenberry polyphenol increased SART-T1 in endothelial cell, and then activation of SART-T1 suppressed apoptosis in the endothelial cell, and then this led to the enhancement of capillarization in the brown adipose tissue, and also the enhanced thermogenesis, and also improvement in the systemic glucose tolerance in the obese, dietary obese mouse. All right, so finally, I would like to talk about the brown adipokine. I apologize, still I cannot disclose the name, but I'm writing the manuscript, that hopefully I can disclose it sometime this year. But the point is that we were interested in finding the adipokine in the brown adipose tissue, like TNF, interleukin 6, MCP, etc., was produced from the white adipose tissue. So what will be the role of brown adipokines in the gabatokine? And then we tested the two microarray data and focus on secreted type profibrotic protein. This increase with obesity or aging. So today I need to call this as OAFP. So we found that OAFP predominantly expressed in brown adipose tissue that would increase in transcript with a high-fat diet. Also in the protein data also indicates that this secreted type fibrotic protein increase in the brown adipose tissue and the dietary obesity. So we also found in the circulation, the OFP would increase in the ELISA, as you can see here. Okay, the point would be obesity, fibrosis, disease with unmet medical needs. And then we came on the NASH, non-alcoholic steatohepatitis, came first as a disease, deadly disease with unmet medical needs. As we know that DM population continuously remains high. And then there's no accurate statistics, but it is said that 37% of the type 2 diabetes patients have non-alcoholic steatitis, like NASH. So the pathogenesis of NASH remains unclear. But still, the molecular mechanism of pathogenesis for NASH remains unclear. But fibrosis progression is in general considered to be one of the main pathogenesis for these deadly disorders, leading to psychosis, even hepatocellular carcinoma. And then therapeutic approach is still extremely limited. So we generated the systemic as well as branch post-tissue OFP knockout model. So this is a systemic knockout model. In the branch post-tissue, of course, they showed a reduction. Also, the liver study showed that the pink signal is a fibrosis. The systemic knockout leads to the diminished fibrosis in the liver. And we generated the gain of function model through AAV in the branch post-tissue. Demonstration of the pro-fibrotic protein increase the molecule in the plasma, and also in the liver, the level of fibrosis will increase, as you can see here. And also non-biased study like ELISA for collagen 1 clearly indicates administration of this pro-fibrotic protein in the branch post-tissue increase fibrosis in the liver. So this is a collaborative study with Dr. Hironori Nakagami, Osaka University, and Shuji Tran in Niigata University. What we found is that the branch post-tissue produce this pro-fibrotic protein, and then this leads to the pathogenesis in non-alcoholic steatohepatitis, as the proof of concept tested in the branch post-tissue specific knockout model, systemic knockout model, and peptide vaccination therapy model, and also a gain of function model. So we tested AAV, and then also we generated a knock-in model. So kind of a gain of function model for a branch post-tissue specific OFP overexpression model, also showing the phenotype. And then in the NASH patients, we found the OFP increase in the circulation. So this is a recent study showing that with aging, we analyzed patients in Juntendo and also in the Niigata University, and we found that with aging, the OFP increase in plasma. And the keyword would be like aging, fibrosis, and then unmet medical needs. And then, of course, like heart failure with preserved ejection fraction is one of the deadly disease. As you know, SGLT2 inhibitor is the only established approach, therapeutic approach for these deadly disorders. And of course, there's several pathogenesis reported, considered to exist in the for the HFpEF. But as you know, the fibrosis is one of the main pathogenesis for this deadly disorders. So with aging, all right, so we among these patients, we extracted the patients with HFpEF, and then in the HFpEF patients, also like aging increase this pro-fibrotic protein in circulation, and this is a study from the EOY prime, like the echocardiography findings. So the high EOY prime indicates the most severe form of diastolic dysfunction. So with a higher E/E prime number, the OFP level increase statistically significant. So we analyze like a systemic knockout model together with branch post-tissue knockout model, and also I got this data like 1 week ago. So we analyze a branch post-tissue knock-in model, and then if you deplete this molecule, the EOY prime will improve in the mouse, normal chow, the high-fat diet, and also in the branch post-tissue specific knockout model in the high-fat diet mouse, the EOY prime would improve, as you can see here. And in contrast, like in the knock-in model, like a gain of function model, EOY prime would increase, as you can see here. So I consider that these data clearly indicates also we successfully generated new HFpEF model in mice. I think the most established model would be from Joe Hill in UT Southwestern, published in Nature several years ago. So we hope this will be contribute for the drug screening or even like herb screening or whatever to generate a new therapy for HFpEF. All right, so the story doesn't end here. So we're interested in like atrial fibrillation patients. That hasn't reached statistical significance yet in humans, but patients with atrial fibrillation have a higher profile for this secretory type fibrotic protein. When we analyze like the atrium for the mice in the branch post-tissue specific knockout model show that reduction in the fibrosis in the atrium. So we consider that the suppression of this molecule will become also the therapy for atrial fibrillation. We are collaborating with several groups in Osaka University, Niigata, and Kyoto University. And in the control group, compared with the control group, like HFpEF patients, atrial fibrillation patients, chronic kidney disease patients, NASH patients, sarcopenia patients, they have a higher profile for OFP, as you can see here. So this molecule increase with aging and also with this kind of fibrotic related disease. So in 2020, I won the National Academy of Medicine award from the National Academy of Medicine USA through this concept. The think aging, the think aging approach, I got interested in the secretory type molecule, like a pro-fibrotic protein in circulation, in mouse, obesity, aging, and also in the humans. So like a NASH, CKD, or sarcopenia, HFpEF, atrial fibrillation, this is going to be categorized like the age-related fibrotic disorder, AFib. And then it's kind of a one syndrome. And then the common pathogenic role of OFP for AFib should exist. And they're pretty much interested in making a drug for targeting this disease with unmet medical needs, targeting the OFP. Right, so the contribution for humans would be enormous. HFpEF patient like 13 million, atrial fibrillation of 13 million, NASH 3 to 5% of the whole population, CKD 10 to 12% of the whole population, and then not many. The therapeutic approach is still unbelievably limited again. And also, like SRD consortium are making AFib research consortium, the kind of global members. If you're interested, I'm really happy to make further discussion and then the kind of a roadmap for making a new disease. And then, for example, like our MBL is collaborating with MBL company, and then screening like a neutralizing antibody targeting this molecule. And also with Daiichi Sankyo, pharmaceutical company, we are now trying to make a high throughput screening for exploration of small compounds targeting this molecule. All right, I'd like to make a summary, like branch post-tissue regulated systemic metabolism and branch post-tissue dysfunction and disorienting coding metabolism from pathologies in failing hearts. And also like think aging approach and OFP contribute for suppression of OFP may become a therapy for AFib, age-related fibrotic disorders. Suppression of stenocytes, stenolysis. I didn't have time to talk about stenolysis. I apologize for that. Specific depletion of senescent cells may become therapies for age-related disorders. Thank you very much for kind attention, and I'm happy to take questions.
Thank you, Shimizu Sensei. Great talk about this. I think it will enlighten our understanding on metabolism of brown adipose tissue. I would like to invite any question from our colleagues here. Please, can post directly. Or any question, you can also post your question on chat if you want.
I think Dr. Patrick Ransom is raising hand.
Okay, please, Dr. Patrick Ransom, please.
Thank you very much. Thank you very much, Professor Smith. It was a very, very interesting and top presentation on broad topics. I'm quite interested in your results on TMAO that seem to increase with aging. Do you think that is because people at higher age have different eating habits, or does the gut microbiome change to produce more TMA, or is the liver producing more FMO to produce more TMAO, or is the clearance decreased?
Yeah, that's a very important point. We're also very much interested in the pathway, why aged individual has a higher expression profile of TMAO. So suppose gut microbiome a dependent pathway as a canonical pathway. We are interested in non-canonical pathway, because when we prescribe antibiotics in the aged mice, level of TMAO doesn't decline. So control group and the antibiotics group, the level of TMAO are pretty much higher, they're similar. And we are interested in another enzyme that would increase with aging, and then the enzyme seems to contribute for the production of TMAO. When we think about the source of choline, like cell membrane has a kind of the source of the choline. So those non-canonical pathway will be pretty much important. We consider with the production of TMAO. But the food-derived TMAO production, of course, the bacterial flora is important, but non-food derived non-canonical pathway, they may be like half important, I guess. So we generated knockout model, and then we have to wait like 2.5 years and see whether the TMA would reduce in those knockout mouse. It's a really painful experiment, but it's kind of a homework, ongoing project. Thank you very much.
Thank you very much.
I think it's clear answer from any more question. Okay, Jeffy, please post your question.
Hi,
Thank you very much. Thank you very much for attending this symposium. Nice presentation.
My great pleasure.
So back to the basic science knowledge that maybe as Dr. Wataru Kimura mentioned previously, that the zebrafish, like a neonatal mice, has a lower body temperature, and this lower body temperature can improve the cardiac regeneration. So it's unlikely has a contradiction result with your clinical results in the heart failure patient. So of course, we should believe the human data compared with the animal data. But what do you think? Do you have some comment about that? Thank you.
Yeah, I don't have many knowledge about the zebrafish, the fisher fish, and then mammalian, the mammalian. That may be the important point, but still we are interested in the regulation of the thermogenesis. What we did in the TAC-operated mice will be, we put them, in the normal temperature. The mice are under 25 centigrades. And when we put the mice in the 14 centigrades, in contrast, we put the mouse in the 30 centigrades, like sauna condition, rather cool condition. But in our hand, there were no phenotype. Even the increase in the temperature, reduction in the temperature didn't ameliorate or improve or enhance pathogenesis and heart failure. Some other group tested like 4 centigrades. It's a really painful condition for mouse. And they say that the mouse die and the heart failure become more enhanced when the mouse are under 4 centigrades. But we didn't test the temperature.
Okay, fantastic. Thank you very much.
Okay, thank you, Dr. Jeffrey. Any more question from our colleagues here? Okay, I have some comment or discussion on this. Simulsense, as you mentioned before, the coagulation would result into whitening, right? More white. Yeah.
Yep.
So in that condition, what kind of effort can be done clinically to preserve this BAT bone adiposis rather than preventing or any bad things happen to our heart would result into whitening, and whether this can be all of this result all this can be yeah, and whether we can have any effort on preserving this BAT. Please.
Thank you very much. That's an important point. So the most safe way to enhance activity of the brown adipose tissue is an exercise. But the point is that it's a little bit difficult to let patients take exercise, right? Including myself, sometimes in Japan it's too cold to go for a run or something. So I'm pretty much interested in making, like similar with others, making kind of exercise mimetics. You know, take the tablet and then skeletal muscle, AKT, just in skeletal muscle may get enhanced. We consider the activation of AKT in the heart, they promote heart failure. But for example, like mimetics for exercise, that may contribute for the healthy brown adipose tissue. And also we tested like a coagulation factor. I mean, we tested the suppression of coagulation factors in humans. And we did some clinical studies, but due to some limited number of patients, we couldn't see any phenotype for the factor 10 inhibitor in humans. And also the recruitment of the atrial fibrillation patients are really becoming difficult because we do not prescribe warfarin. So it's going to be like a single arm study, just factor 10 inhibitor, yes or no. You know, we cannot make a comparison. Thank you very much.
Okay. Professor Patterson, do you have any more comment on this, please?
Maybe I can comment on this also, because what we also see, for example, in mice, that with aging, brown fat gets more white. And we see that there is a very high diurnal rhythm in the brown fat activity, with a very high activity at the beginning of the wakeful phase. And we've also seen last year in humans that the ability of a human being to increase energy expenditure to cold exposure is higher in the early morning and in the late afternoon. So I have the feeling that if we can sort of preserve the natural biological rhythm, and I'm not really sure how to do that, but with aging, of course, the biological rhythm decreases. And if we have some idea to have the biological rhythm in place, then we can also maybe prevent whitening of brown fat in mice, but also in humans.
Yeah, I agree. Yeah, there's so many things to be done.
Yeah, absolutely.
Yeah, I think this also open up for future study and more how to preserve this BAT to become whitening, right? So, and any more question, answer, or discussion from our please? Dr. Bambang, you have any idea? Discuss on this?
No, but I enjoyed the lecture from Professor Shimizu. Thank you so much.
Thank you very much.
Okay. If no, I just okay. Again,
Yes.
Please.
Yes, maybe one quick question. So, regarding to the quantity or the quality of the endothelial or the vascular in the older adults population. So you mentioned that the decreasing of the angiogenesis can become a detrimental effect in older adults, but it's like in the cancer patient, that's that angiogenesis is decreased in the cancer patient, but in fact that the older adult has a higher prevalence of the cancer patient, so it's kind of a contradictory results. So what do you think? Or maybe the quality of the endovascular is rather important than the quantity. Thank you.
Yeah, that's an important point. So when we think about the maintenance of the vascularity, I mean the capillary network, there is to be at least two approaches. One will be the enhancement of VEGF, another will be the suppression of end cell death. So initially, I extensively studied on the VEGF, like HIF1 pathway in the heart on brown adipose tissue. But some type of the cancer, we use a VEGF inhibitor. So there, I think in general, the VEGF activation has a kind of a promotion, the risk for the promotion of the cancer. So I'm not pretty much interested in the VEGF regulation, but instead a little bit focus on the maintenance or suppression of EC cell death, including like, for example, the ROS suppression in EC will lead to suppression of cell death in EC. And also, for example, the senolytic approach, the depletion, the lysis of the senescent end cell may enhance the proliferation of end cell. So I will be making a shift from VEGF to the EC, suppression of EC cell death, including the renewal of the EC. So I think we can, that approach will be more, safe approach when we think about the cancer. Yeah, that's important. Interesting question. Thanks, JP.
Thank you.
For that.
Thank you very much.
Thank you, Dr. Jeffy. Thank you, Dr. Shimizu Sensei. Any more questions from colleagues here? All right. Due to our limit time, and we'll hear another more interesting and more experts here. We'll hear together. I would like to thank Professor Shimizu, Dr. Shimizu Sensei, Dr. Ipe Shimizu for these great lectures. Give us enlightenment on the one things that it can be our way to do something in terms of improving cardiovascular and metabolic health by seeing brown adipose tissue as the one things, and the way we maybe in the future we have need more study on the mechanism of action and how we can preserve this adipose tissue, especially the brown one, and also to prevent the whitening process. For this, I'd like to thank everyone for the great contribution and also the discussion, and thanks for our committee for inviting us for this session, and hopefully everyone can enjoy this great meeting. Thank you very much, and I'll give back to Natasha. Thank you.
Thank you very much.
Thank you very much, Dr. Ipe Shimizu, for the very interesting presentation. And thank you as well, Dr. Renan Sukmawan, for leading the session. Very well, ladies and gentlemen. I hope all of you are enjoying the event so far. Next, we will have a short break for 10 minutes. For the participants who want to go to the restroom or stretch, you can utilize this break session. We will be back for the next session of CMARS DA1 after the break. Thank you very much. Good afternoon and welcome back to CMARS 2023. We hope that you had a sufficient break time because our next session will be just as engaging as before. Our next session is Meet the Editors, What Editor Wants by Nature Journal. That will be led by our moderator, Dr. Bambang Widiantoro, and we also have a panelist, Dr. Sunu Budi Raharjo. Good afternoon, Dr. Bambang and Dr. Sunu. I hope both of you are enjoying our event. But before we continue the session, allow me to read the CV for Dr. Bambang Widiantoro as the moderator for all of the participants. Dr. Bambang is from the Department of Cardiovascular, Faculty of Medicine, University of Indonesia. He had his medical doctorate degree in University of Indonesia, and he is a graduate of Kobe University and a cardiologist of University of Indonesia. His activities consist of a deputy editor of Indonesian Journal of Cardiology, associate member, writing center cluster of IMERI, Faculty of Medicine, University of Indonesia, principal investigator of Cardiovascular Division and Metabolic Disorder, Cardiovascular and Aging Cluster, IMERI, lecturer of Department of Cardiology, Vascular Medicine in University of Indonesia, and intensive care medical staff and cardiovascular emergency, National Cardiovascular Center Harapan Kita. Very well, without further ado, I will pass the spotlight to Dr. Bambang Widiantoro. To the doctor, the floor is yours.
Thank you so much, Natasha. Very warm welcome to all of you. Thank you for joining the session. Welcome to our fruitful session. So this is one of the session that I always want to join in such a conference, since not every medical and scientific conference has session to meet the editors. And today we are pleased to have our distinguished speakers, Dr. Yanina Yasmin Pesk. How are you, Dr. Pesk?
Hello, I'm fine. How are you?
Fine, thank you. Thank you for joining us today.
Yeah, thank you so much for the invitation to participate and present the journal. So I really appreciate the invitation.
Yes, we would love to have you here. And we also have Dr. Sunu Budi Raharjo as our panelist. So I would like to first, can the organizing committee show the curriculum vitae of our speakers and panelists, please? Oh, Dr. Pesk, your cat is so cute.
Yeah.
Okay, so Dr. Yanina Yasmin Pesk is currently the associate editor of Nature Metabolism, one of the very prestigious journal in the metabolism field, and she earned her master in University of Bonn, Germany, and also PhD in University of Bonn and also Leipzig. And she had her postdoctoral fellowship in University of British Columbia, Canada. And I believe you already joined the Nature Metabolism as an associate editor from last year, I think.
Yeah, I did.
So
I've been an editor for exactly 1 year. So exactly 1 year ago on this day, I moved back to Germany to start the new job.
Oh,
so
great.
It's a nice way to celebrate this anniversary.
Yeah. Thank you so much for joining us. And
hello, Dr. Bambang. Dr. Yanina.
Hi, Dr. Sunu. How are you?
Good. Good afternoon.
Dr. Sunu is my senior in Department of Cardiology and Vascular Medicine, University of Indonesia. He earned his medical doctor from University of Gajah Mada, and then PhD in Kobe and cardiologist from University of Indonesia. And currently he served as editor-in-chief in the Indonesian Journal of Cardiology, the official journal of Indonesian Heart Association. And also he was the research coordinator in our department and also staff member in our National Cardiovascular Center Harapan Kita Hospital. Welcome and thank you for joining us, Dr. Sunu.
Yeah, thank you, Dr. Bambang. Thanks for the opportunity. Actually, we should revise the CV. The research coordinator is Dr. Bambang, right? Hello, Dr. Yanina. So nice to see you. Yeah, looking forward to learn from you. Yeah.
Okay, so please, Dr. Pesk, to deliver your presentation, and we look forward to enjoy it.
All right, just give me a second to share my screen. Can you see my slides now?
Yes, perfectly.
Perfect. Okay, so then I'll get started. So thank you so much for the invitation to speak here today. So I'm Janina Pesch. I'm an associate editor at Nature Metabolism, which is a Nature portfolio journal. So you already heard a little bit about my background, just going into a little bit more detail. So I studied molecular biomedicine and life and medical sciences. My PhD was actually in developmental biology, not in metabolism. And then I spent almost 5 years in Canada doing postdoctoral research at the University of British Columbia. And as I already said, exactly a year ago, I joined Nature Metabolism, and I'm based in Berlin. So first of all, introducing the journal Nature Metabolism. So Nature Metabolism is an online-only journal of the Nature portfolio. It was launched in January 2019, so it's a relatively new journal, and we have 12 issues per year, which are online only. So our team consists of 5 editors, with me, our editor-in-chief, Christoph Schmidt in Berlin. We have another editor in Berlin called Alfredo. Then we have Ashley in New York and Isabella in London. And among the team, we have certain topics kind of distributed. So for instance, Alfredo handles all the manuscripts related to cancer metabolism and immunometabolism. I handle genetics, microbiome research, and all the manuscripts that are very signaling heavy. Then Ashley handles neurometabolism, nutrition, and exercise metabolism, and Isabella handles everything related to inner organs like liver and pancreas, adipose tissue, and so on. So yeah. Then what do editors do? So we filter the manuscripts. We always say that we read 1,000 manuscripts, so you don't have to, because we only select the most interesting ones, and all the manuscripts we receive are kind of like diamonds in the rough. So with our work, by guiding the peer review process and the revisions, we enhance the content of those papers so that from the diamond in the rough, we can get this nice sparkling diamond. And then by giving the authors a platform to publish in our journal, and by spreading the word on social media and highlighting papers and press releases, we also amplify the message and those manuscripts. So how do editors spend our time? So of course, most of what we do is handling manuscripts at all stages of submission, from new submissions to manuscripts that have already undergone the peer review process and are close to acceptance. So a lot of manuscript handling, but that's not all we do. We also spend a lot of time keeping on top of the field. So that can be reading papers published by other journals, but we also do a lot of outreach, in which we contact researchers or we talk to researchers about their ongoing projects. We also attend a lot of conferences. So as you can see on the right, in 2021, so these are still pandemic numbers. This number will only grow now that most of the restrictions are lifted. The Nature portfolio editors, so not just Nature Metabolism, but all Nature journals, made over 1,000 lab visits, attended over 600 scientific conferences, and gave 161 presentations. So that's also a lot of what we do, and that's also something that we all really enjoy. So now, Nature Metabolism, what do we publish? As the name suggests, we are very interested in metabolic research, but we have kind of a broad scope. So we publish everything from metabolic cell biology and biochemistry, so in vitro studies, to translational and clinical research like human clinical trials and so on, which is also something that we really want to grow in. But most of the research that we publish is preclinical research in metabolic physiology and biomedical research. So for instance, some work in isolated tissues or animal models, everything from worms to flies to mice. Most of the research that we publish has been done in mice. So our scope is relatively broad. So now what makes Nature Metabolism special? Okay, my cat just wants to say hi and introduce the journal. So we aim for fast decisions. In comparison to other journals, we try to be very fast. So when you first submit a manuscript to us, we try to get back to you with a decision on whether or not to send this out for peer review within 7 days. And if we send it out to review, we want to try and make a decision on whether or not to invite a revision after 40 days. And also when we reject a paper, we always give you feedback. We don't reject it right away, but we write you the points that were missing or why we rejected this paper. And if we invite a revision on a paper, we also give an editorial guidance going through all the peer review reports and filtering out the points that are the most important for the journal and that you should definitely pursue in a revision. So we try to help the authors at every stage. And then, like many Nature journals, we offer double-blind peer review options. We have implemented the Nature journal standards for reproducibility. And we can also promote articles via press releases. And we are a transformative journal, which means that in addition to the standard subscription model, we also have open access publishing options available. So now, what does Nature Metabolism publish? We don't just publish research papers, which are in like the back half of our journal. So those research papers would be letters and articles, but we also have a front half, which is commissioned content like review articles, perspectives, which are like shorter reviews, comments, which are more opinion pieces. We also feature editorials. Metabolic messengers are like mini reviews that focus on one specific metabolite to give a nice overview. And just giving you a couple of examples. So editorials are very short pieces where us editors talk about some subjects that we find important for the community. And those can vary a lot in topic. Then we have a lot of review articles and perspectives, and we put a lot of effort in those, where we ask the leaders in the field to discuss broader topics like cholesterol metabolism or a metabolic handbook for the COVID pandemic and so on. So yeah, we also occasionally have focus issues, so kind of special issues where we group together certain articles about a certain topic, like we just had one on neural control of energy homeostasis. But for instance, for the last Olympic Games, we had a collection on exercise metabolism. And yeah, we recently had one about improving reproducibility and reporting standards, where our goal is to provide some guidelines for certain analyses or reporting standards in the field, which will hopefully solve some common problems in the field in terms of methodology and reporting. And then we also feature career pathways, where young professors that had their first major last author paper in Nature Metabolism talk about their path to academia. So those are quite interesting. And for the research highlights, we don't just publish the standard metabolism manuscripts. Of course, we publish a lot of them, but we also occasionally publish some really odd papers that you would not think make it to Nature Metabolism, but they do. So we are not afraid of publishing negative results. And for instance, we also publish manuscripts that deal with flies or worm models, or we even had a really popular one on squirrels. So yeah, our scope is pretty broad. So now I want to talk a little bit about the publishing process and along the way give you some advice on how to make your manuscripts a relatively strong contender for peer review. So you're probably quite familiar with the flow of a manuscript. So you write it, you submit it to a journal where the editors make an initial decision on whether or not to reject the paper or to send it out for peer review. In which case the referees would give their feedback to us editors, and then us editors make the decision on whether or not to reject the paper because it was not well received, to invite a revision, or whether to accept the paper because it was exceptionally well received. So first of all, when you're preparing a submission, you have to ask yourself the question, how big is your story? If you've made a great discovery that will have a lot of implications for research, then you might actually want to try a journal like Nature first instead of submitting it to Nature Metabolism straight away. Then if you only have a very short manuscript that doesn't feature a lot of results, then it will be hard to get into those bigger journals, and you might want to try another journal first. Then also one question is, what audience do you want to reach? Do you want to reach a broad audience? Then you can try one of those broader scope journals. Or do you want your research to be accessible to your specific subfield? Then you might want to choose a field-specific journal. Also, one question is, how fast do you want to get it out? For instance, if a student has to graduate, then going for Nature or a Nature Metabolism might be a bit difficult because it will take quite a while to undergo the largely extensive revisions that higher impact journals usually ask for. But if you try a smaller journal, then the revision process will be relatively easy, hopefully. And then, because not all journals offer open access options, another question is whether open access is important to you. So we currently offer an open access option, but also a non-open access option, whereas other journals are fully open access and some don't have any open access at all. So when you are preparing your paper, then you have to try and get the main message across. That's the most important thing. So we don't care about the initial formatting of your paper, so that we don't care about this, but it has to be well written. And one thing is also, even though we don't care about the font size or whatever, we want you to familiarize yourself with our editorial and publishing policies, which are available on our website. We offer double-blind peer review options, and if you are going for that, you should write the paper in a way that does not disclose your identity. And also, please include a cover letter with your submission. So I'm not going through this whole list of tips, but one thing that is important is sex and gender reporting for all animals and all human participants of trials and so on. So that is something that the journals are all increasingly looking out for. Then we also are looking at statistics. So some tips are, for instance, using scatter plots instead of bar graphs and listing all statistical tests and all n numbers and all the figure legends. And because we will be anyway asking for it in a later stage right before acceptance, it should be easy for you to make your data available. So when you're actually working on those projects, please keep all the source data because most journals will be asking for it in the Nature family. And also try and make your code available as early as possible. And then also one important point is ethics information for research on animals and on humans. And we also have specific guidelines for clinical research, which you can find on our website. And then once you have submitted the manuscript to us, the handling editor will guide you through the editorial process. So as I already said, different editors at the journal handle different topics, but sometimes there can be some overlap. And we also often discuss about manuscripts. So we usually don't make the decisions on our own, but as a team. And we try to get back to you within a week. of submission. And what are we looking for? So all the points listed below are actually something that all journals are looking for, but different journals weigh the different points differently. So for instance, if you want to publish in Nature or Cell, for example, all the points below have to be addressed really well. If you publish at a small journal, they might not really ask for all these points to be addressed super well. And yeah, every journal has different standards, but in general, what we're all looking for doesn't really vary that much. Just the different degrees of how we weigh the different points varies from journal to journal. So what is important for us is relevance to our readership. So if you submit a manuscript that has nothing to do with metabolism, then it's not a great fit for Nature Metabolism and you should probably try elsewhere. Then the significance of the finding and the level of conceptual advance is very important to us. So the higher you go in impact factor, the more important that point is for the journals. And also the conclusions that you made in this work should be strongly supported by experimental data. And then depending on the type of paper, different considerations are important. So for instance, for biochemical work or like more mechanistic studies, we need a lot of mechanistic insight and physiological relevance. For studies that address work in like mice or flies or so, we usually want to see some relevance to human physiology or disease. There's also certain papers that don't give a lot of mechanistic insight, but have a great resource value, but we would also, of course, consider those. For methods papers, the technological advance has to be great. And of course, if you would submit to a methods-only journal, the standard there would also be really high. And depending on the paper, we could also ask for societal relevance, even though for Nature Metabolism, that's quite rare. But yeah, those are the most important points. And I would say the top ones, significance of the finding and conceptual advance, strongly supported, well done experiments, and physiological relevance are probably the most important to us. But as I said, that depends on the paper. So a strong contender for peer review answers an important question in the field, has data that is new and interesting, the experiments are well done, and the conclusions are very well supported by the data. So now your paper has ticked all those boxes or a lot of the boxes, then we send it out for peer review, and the reviewers, which are independent experts in the field, address whether those points are well done, the conclusions are supported, the data is of high quality, statistics and methodologies are fine, and they also give us some insight into whether or not this is an appropriate scientific advance of the field, or if there's any overlap with previous work, which would dampen our enthusiasm for the study because we want to publish things that are novel. So then we receive those reviewer reports, and us editors then make the decision, and we don't count votes. Like 2 reviewers were happy with the study, 1 was negative, so we actually invite a revision. If the negative reviewer had some very, very important arguments and discovered a fatal flaw in the manuscript, then we still can't invite a revision. So we consider arguments, and we also occasionally overrule reviewers if someone is asking for experiments that would take several years or so to address. And we use our own judgment on which of the requests are feasible. And we really try to uphold very rigorous standards for review. So once we have invited a revision, one thing that is really important is that you make the most of your opportunity to revise. So you can't just say, no, I'm not interested in answering this point by reviewer 1 because reviewer 1 is an idiot. That's not professional. And we also really want that all experiments are addressed. All the points by the reviewers have been answered, because if this has not been answered, then the reviewers will definitely also complain during the next round of review. And also what we often do is that we discuss revisions with the authors. So that is something that not many people are aware of, but we actually offer this service, and a lot of researchers pick up on that. So if you have a question about a revision, then it's totally fine to talk to the editor. We want your paper to get published. We don't want to reject your paper unless we have to, but we want to help you. And one thing that is quite important is that you also write a really extensive and professional point-by-point response. And for instance, if one of the points could not be addressed experimentally, then also you need to explain this really well. So in case your manuscript got rejected, either after peer review or after the initial submission, then we can still initiate a transfer to another Nature portfolio journal. For instance, a lot of the manuscripts that we reject then get published in Nature Communications, which is also a great journal. So we can consult with our sister journals to see if they would be interested in your manuscript, even though it does not really meet the bar for Nature Metabolism. And the other way around, some papers that got rejected from Nature then get transferred to Nature Metabolism and get published with us. So there's always an option. But hopefully your manuscript will then get accepted. Then it still takes a couple of weeks until it actually shows up online because of all the formatting and form filling and so on, sub-editing. We are always happy to promote new accepts via press releases or news and views, which are highlighting overview articles. And also we share your manuscript on social media once it got accepted, and you can also, of course, share your work. So yeah, so that was the publishing process of Nature Metabolism and what we are specifically looking for. And if you have any questions about this, feel free to ask them in the Q&A session or also contact me. Or if you have any projects that are close to completion and that you think would be a good fit for the journal and that you would like to discuss, you can always contact me or the other editors. Yeah, thank you so much.
Okay, thank you so much, Yanina. It's a great presentation. I enjoyed it so much, and I believe the audience as well. So I would like to give the first opportunity to Dr. Sunu to give some comments and questions to Yanina.
Thank you, Dr. Bambang. Thank you, Yanina, for your excellent tips and tricks for publishing, and especially in Nature Metabolism. I'd like to give comments as an editor as well as author. You mentioned that in Nature Metabolism, there is one chief editor, and then for associate editors. What is the process of acceptance of the paper? I mean, one associate editor is has an authority also to do acceptance, or should be decided by all those editors, so the chief editor and the 4 associate editors?
So the chief editor has the last say. So all the decisions that are post the initial peer review process are anyway made in the team. So it's always one handling editor, one of us 5 in the team, handling one paper from start to finish. And only the first decision, whether or not a paper can be sent out for peer review, is occasionally made by a single person. But all decisions afterwards are made by the whole team.
Okay. And after we got the review from the maybe 3 reviewers, and we see the content of those reviewers, who will decide the acceptance of the paper? So associate editor is okay, or shall go to the chief editor in Nature Metabolism?
As I already briefly mentioned, so the chief editor has the last say. So if we all don't really agree, then it's up to the chief editor. But usually it's quite clear from the reports on whether or not so if we accept it, then usually there's only very, very minor changes that the reviewers are still asking for. If not, we invite another revision.
Okay, thank you. Can I have another comment, Dr. Bambang?
Of course.
Yeah. And yeah, it is very interesting that you mentioned that during the 2021, the editors have over 1,600 visit to the lab. So it is a whole, I mean, how many hours a week you work in this nature metabolism actually?
Yeah, so it's full-time job. So at least for me in
yeah.
Germany here, it's 37 hours per week. But so those statistics were not for only Nature Metabolism. Those statistics
see.
were for all Nature journals, so like 100, 150 editors. But yeah, we try and talk to authors via like Zoom calls or so at least every week. And then lab visits are usually happening when we are going for a conference abroad. And then we also visit some labs that are close by and that we usually would not be able to visit. And yeah, it's nice to actually talk to people in person. And I'm glad that we get this opportunity now that most of the COVID restrictions are lifted.
Oh, okay. Well noted, Yanina. So Dr. Bambang, it should be full work.
Yes.
So the problem for us, Dr. Yanina, is so me and Dr. Bambang is the editors for Indonesian Journal of Cardiology, but most of our time actually used for working as physicians, and it's very tough to do the same time for editing those many manuscripts. Yeah. So do you have any tips on this?
Yeah, so I realized that it's a very difficult situation. So for all the editors in the Nature family and also at Cell Press, those are full-time editors who don't have a job as a physician or researcher on the side. So we can really focus on handling manuscripts for the entire week and like for our entire work hours. But yeah, so I understand that it's very difficult. And I honestly don't really have that much advice. But yeah, I feel like every manuscript deserves to have some time put in it, and especially the good ones. So one thing you can maybe do is to prioritize. If you see that a manuscript is not that great, you can immediately decide to not really spend hours and hours evaluating it. So maybe that would save some time. And for the good ones, maybe also not spend all that much time looking at it because you trust your gut feeling and think it's great, so you can immediately send it out for peer review. One thing that also helps is for potential reviewers to have like a database of potential reviewers, so you don't have to Google them all the time, but you can just look at the database, which saves some time. But I very much admire everyone who has a full-time research or medical job and then still works as an editor on the side. So
very
that's
tough.
absolutely
Very tough.
great.
Yeah, one of our problem is to find the reviewer actually. Do you have any problem on that?
Yeah, so it always depends. Like in the summertime, it's very hard to find reviewers because at least in Europe, everyone is on vacation. So one thing we do is we increasingly try to involve reviewers from countries like China or like Southeast Asia and so on. So people who are still a little bit underrepresented in research, but we also want to give them the opportunity to review manuscripts for us. So that definitely opens up a whole new pool of reviewers. We should not just focus on those from the US or so. And yeah, in general, it always depends on the different subfield. For some fields, it's easier to find reviewers, for others, it's not. But yeah. Also, one thing that we do is if we invite reviewers, we ask them to give suggestions in case they cannot complete the review. And then they would suggest some other people who they think are great and who can fulfill the reviewing duties. And that's also often junior researchers who we, as a relatively high impact journal, don't really have on our radar, but they can make great reviewers.
Okay, thank you. And I think, I still have many things to ask Dr. Bambang, but I believe there are many attendants who also want to ask to Yanina. So that's for the time being from me. Thank you. Thank you, Yanina.
Thank you, Dr. Sunu. I will appreciate to the audience. I will represent as an author, Yanina. So some of our participants might be an early career investigators or final year master or PhD student or cardiology or internal medicine resident in training, who will have to submit their thesis to the journal as one of the requirements for graduation. So you already mentioned the first tips how to choose a journal. My question is that when we feel that our manuscript and our study probably is good enough, but not great. So in this case, do you think it's good to submit to the highest level first to get the comments and the question from the editors and reviewers, so we can improve our manuscript, even though we might not, we may reject it at the end, but we can have such a good, valuable comments first, or just directly choose the appropriate one due to the quality of the study. What do you think?
So I would always try to aim a little bit higher than you think where your manuscript actually is at. So if you have a very small manuscript, then you would literally only waste your time submitting it to Nature. And some of those very high impact journals also don't really give any feedback on why they rejected your manuscript. We do this, but not every journal does this. So one thing you can also do is to submit pre-submission inquiries to the journal. They are usually handled within a few days, and then you get some feedback based on the abstract of the study, whether or not the editors think that it's suitable for their journal. Or you can contact the editors directly via email and ask them and give them as much information as you want to. And then if we are interested in a project but are not quite sure on whether or not it's a good fit, we also occasionally have some Zoom conversations or email conversations to discuss the manuscript. But yeah, if you think that your manuscript is actually a good fit, then why not submit it? Most of the journals, at least in the Nature family, try to be really fast, and worst case scenario, you would only waste a week or two. But of course, it's very frustrating, especially for the junior authors. If you first try Nature, then it gets rejected from there. Yeah, then you try Nature Medicine, it gets rejected again. It ends up with us at Nature Metabolism, we reject it, and in the end, it ends up somewhere else. And yeah, so you always have to be a bit realistic, but try to aim just like 1 or 2 journals above where you actually think your manuscript will end up with.
Okay, I think that's a good tips. Thank you. I just realized that you have such a method or a way that we can discuss with the editors.
Mm-hmm.
Is that with all of the Nature Family Journal?
I mean, not every journal offers this, and it really depends on the editor's discretion and how much time we have. So for instance, if you're asking to discuss a manuscript right before Christmas, when our office will be closed for a week and we are drowning in submissions because everyone wants to get their manuscript out before the end of the year, then that might not be the best time to discuss a manuscript with us. But we always try and discuss at least the ones that we think are very interesting. And we, as I showed this graph in the beginning, where only like 60 or 70% of our work is actually handling manuscripts. So we have quite some capacity to discuss projects with us. Of course, we can't do a Zoom call for every pitch that we get, but at least you can write us an email. That doesn't really cost us editors that much time to reply.
Okay, thank you so much.
Dr. Bambang,
So,
a few comments. I just realized that the season is important. Like winter, like summer. To find review is difficult during summer, and to contact the editor is not during the winter, something like that. Yeah. So I think we should note that, Dr. Bambang. Yeah.
Yeah. It's important for like Yanina or some of the editors that based on Europe or the US, I think. I think can I have one comment from the audience, please?
Can I have Dr. Bambang?
Oh, sure, Dr. Renan, please.
Yeah, Yanina, it's great to follow your lecture. As an editor, right? So when you receive manuscript for the first time, which part of manuscript you will read first, and you have like any educated guess or suggestion, it could be accepted, it could be interesting for our readers. So then you will decide this paper will go into an editor. So can you tell us what kind of trick? I think you receive lots of manuscript every day, all right, or every week. And when you see it for first time, which part we believe that all parts is important, right? And which part you will see first, and you have the gut to say that this kind of manuscript probably would be interesting or will be accepted or interested for the reader. Then you decide for this manuscript to send or to an editor.
So all the manuscripts are being evaluated by editors, and we always read the whole thing, and then it depends on how good it is or not to actually send it out to the reviewers. But all manuscripts are sent to us editors. But the most important thing that gives us a first overview is the abstract. So I would really suggest putting a lot of effort into writing the abstract, because it sums up not only what is already known in the field, but it also shows what you did and what the new findings are and what the implications are of your work. So that is a really nice overview. Of course, we sometimes get manuscripts with terrible abstracts that end up being great, and we send them out to review, or the other way around, people kind of over deliver in the abstract, and then the actual results are not very great at supporting their conclusions. So it can be any way around, but the thing that first catches our attention is the abstract.
Okay, and you know, probably in some countries their English not, because they're not mother tongue, right? So would you directly go into part of abstract to the results rather than seeing some sentences in the part of introduction? Or so how important you directly seeing the results of abstract?
So the results, of course, the results part is the second most important part, I would say, and you should really put a lot of effort into describing the results and also making some figures that really show them very clearly. So the figures are important too, but I would say they are part of the results section. And so a lot of our submissions are actually from non-native speakers, and none of our editorial team members are native English speakers. So I would not be very concerned about that. Of course, we have to understand what you mean, and it really doesn't hurt to have a native speaker proofread your manuscript before submission, because if the English is really bad, then it's sometimes hard to understand the results. But we don't care if there's a few grammar mistakes or typos or so. That is something secondary, and that will be fixed later on before acceptance in the copy editing stage. Or sometimes if it's really bad, we suggest some languages services to the authors. But don't worry about being proficient in English. As long as we can clearly understand what you mean, it should be fine.
Yeah. Okay. All this information encourage all of us here to more sending more our works into the journal. Thank you.
Thank you, Dr. Renan. And I think we coming to the end of our session. So I would like to again thank you so much to Yanina and also Dr. Sunu for joining us today. And I believe our participants have a lot of broad view about how to choose a journal and some tips that already shared by Yanina. Because as a clinician or a scientist, after we do all the effort to do the study, and then it will not be complete without exchange and dissemination of the result to the public, and it only can be done by publish and submitting our manuscript to a good journal. So thank you so much again, Yanina, for the tips, and hopefully we can meet in another session again. Thank you.
Thank you.
And with that, I conclude our session. Thank you so much and have a good day all.
Thank you. Bye-bye.
Yeah. Thank you for having me. Bye.
Yeah.
Thank you. Send my regards to your cats.
Thank you very much, Dr. Yanina, Dr. Dr. Sunu Budi Raharjo, and Dr. Bambang Widiantoro for the wonderful session. Now, ladies and gentlemen, we will now have reached the last session of today's event, which is the Meet the Editors edition 2, What Editor Wants, Frontiers Journal. This section will be moderated by Dr. Agus Rizal Ardi Haryandi Hamid, and we also have a panelist, which is Dr. Nafrialdi. Before we start, allow me to introduce Dr. Agus Rizal to our audience by reading his CV. Dr. Agus Rizal Ardi Haryandi Hamid is from Medical Faculty Staff at Department of Urology, Cipto Mangunkusumo Hospital, Faculty of Medicine, University of Indonesia. His activities consist of Chief Editor of Medical Journal of Indonesia, Associate Member, Writing Center Cluster IMERI, Faculty of Medicine, University of Indonesia, Medical Staff, Department of Urology, Faculty of Medicine, University of Indonesia, Secretary of Urology Study Program, Department of Urology, Faculty of Medicine, University of Indonesia, SIU Innovator Committee, Society International de Urology, and board member of Asian Urological Surgery Training and Education. Without further delay, to Dr. Agus Rizal Ardi, the time and place is yours.
Thank you very much for a kind introduction. Can I start it? Hello?
Yes, Doctor, you may start.
Yeah. Okay. Thank you very much for your kind introduction, moderators. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. So we're going to have a nice session now. We are going continuing the previous session. Recent session is will be discussed about what editors wants, especially in the Frontier Journal. We know actually in the last couple of weeks that we have sort of like a debate about the Frontier journals in Indonesia, but I think this is the good times also. We going to learn about the Frontier journal. In this session, we have two speakers. Can I have the CV? Can we show you the CV? Okay, the first speaker will be Sundaram Arumugam, PhD. He works in Department of Pharmacology and Toxicology, National Institute of Pharmaceutical Education and Research, Kunilal Bawan. Sorry, maybe I make a mistake during pronunciation. He had a degree, bachelor and master in Chennai, and PhD in Niigata University of Pharmacy Applied Life Science, Japan. Now he's also associate editor Frontiers in Pharmacology, and assistant professor in NIPER, Kolkata, and many fellowship, and also got the award for academic excellence from NIPER, Kolkata in 2019. The second speaker will be Professor Wawaimuli Arozal, MD PhD. Now she is working Department of Pharmacology and Therapeutic Faculty of Medicine, Universitas Indonesia. She also now become Associate Editors Frontier in Pharmacology and Research in Department of Pharmacology and Therapeutic Faculty of Medicine, Universitas Indonesia, and staff and associate member in Metabolic Disorder, Cardiovascular, and Aging Cluster, IMERI, Faculty of Medicine, Universitas Indonesia. And we're going to have one panelist that will give more comment and insight. And we already know about Dr. Nafrialdi, MD, PhD. He works also in the Faculty of Medicine, Department of Pharmacology and Therapeutic, Faculty of Medicine, Universitas Indonesia. Actually, he was a past editor-in-chief Medical Journal of Indonesia before me. Now he's still as an editorial board member, and also he is editorial board member in Acta Medica Indonesia, and he also have a lot of experience in become editor. Now he also works in the Secretary Research Ethics Committee, Faculty of Medicine, Universitas Indonesia. So first, I will give time to the Soma Sundaram Arumugam. Sorry, I have to pronounce very slowly, otherwise I get the mistake in pronunciation. So, Mr. Suma.
Yeah. Very good afternoon, everyone.
Hello. Hello. Yeah.
Audible?
Yes, we can hear you clearly, but I think it's maybe it's a little bit louder or something. Because still the sound is quite.
Is it fine?
Yes, now it's fine.
Okay. Okay. Yeah.
Good. Thank you. So please.
Yeah, first of all, I would like to thank the organizing committee for giving us this opportunity to maybe discuss about what we are looking in frontiers in pharmacology. Frontiers, as you know, has many kinds of journals in which me and Professor Vavai Muli both are associate editors. So I would like to hear from Dr. Vavai Muli also. Muli-san?
Sorry. So you can share your PPT first, and then I will add some information later on this.
Yeah, thank you. So can you see the screen?
Yes, yes.
Yes.
Yeah. So just I would like to introduce like what are the different As Yanaina has clearly explained what they are looking in Nature Metabolism journal. That is how the submission process and the review process and acceptance or rejection, all these things she has detailedly explained. But here I will explain about some parts of the manuscripts as well as different ethics or guidelines what we are following in frontiers of pharmacology especially. So first and foremost thing is the different types of manuscript. Whatever the manuscript we submit, the authorship and author responsibilities, that is most important thing. Like what frontiers follow, they follow the International Committee of Medical Journal Editors guidelines, that is ICMJE guidelines. Excuse me. Yeah, sorry for that sound. Yeah, so we follow that ICMJA guidelines, which state that in order to qualify for authorship of a manuscript, the following criteria should be observed, like substantial contribution to the conception or design of the work, or the acquisition, analysis, or interpretation of the data of the work. and drafting the work or revising it critically for important intellectual content, and provide approval for publication of the content. And finally, this foremost important thing is agree to be accountable for all aspects of the work in ensuring that questions related to the accuracy or integrity of any part of the work are appropriately investigated and resolved. So all the authors whoever the author of a publication, they should take the responsibility that any question or any query arise on the accuracy or integrity of the manuscript or the part of research, they are all responsible for that. And the contributions, like it should be clearly mentioned to provide appropriate credit to all the authors, as well as assigning responsibility and accountability for published work. Individual contributions should be specified as an author contribution statement. Like example, you can say different authors. We have to give only the initials like A, B, C, Z, D. They contributed to conception and design of the study, and who organized the database or performed the analysis or wrote the first draft, or finally, all authors contributed to manuscript for revision, read, and approve the submitted version. So these statements must be given in all the manuscripts. And as we all know, corresponding author will be the direct communication or contact point for any query related to the particular manuscript. He is the responsible person. The corresponding author should be responsible for ensuring that the submission adheres to all journal requirements, such as details of authorship, study ethics, and ethics approval, clinical trial registration documents, and conflict of interest declaration. So the corresponding author should also be available post-publication to respond to any queries or critics. And a request to modify the author list after submission should be made to the editorial office using the authorship change form. So these are some guidelines related to corresponding author. So now we are coming to the research integrity and ethics in publication. So original content and duplicate publication. This is most important thing, where whatever the manuscripts which are submitted to Frontiers must not have been previously published or be under consideration for publication elsewhere, either whole or in part. So if an article has been previously submitted for publication elsewhere, so Frontiers will only consider if the article has been definitely rejected by the other publisher at the point of submission to the Frontiers. It's not like it's already under communication in some journal and you can submit to Frontiers. So this is common for even any other journal also. And regarding the data or images, so fabrication and falsification, this is the most important thing we have to consider. Otherwise, the manuscript may go for rejection or retraction. So Frontiers opposes both fabrication of data or images. So that is, what is fabrication? That is fake or made up data. Similarly, falsification of data or images is manipulating the data or image so that it is leading to a misrepresentation of any data. So these are clearly not accepted in Frontiers. And the redundant publications, like submissions which are highly related, are very similar to articles which are already published, or the similar kind of experiments or study material or unethically conducted. All these things are not considered. And the plagiarism, which now almost all the journals they follow this early check, the preliminary check using various tools like Authenticate. That is also followed by Frontiers. So in accordance with that COPE guidelines, the publication ethics guidelines, so we expect that original wording directly taken from publications by other researchers should appear in quotation marks with appropriate citations. So this also applied to one author's own work. If it is that is called a self plagiarism. So this all should be avoided so that the rejections can be prevented. So whatever the data which are already presented in conferences, proceedings, or abstracts, so they can also be published, but the authors are required to add a substantial amount of original content in the form of new raw material or experiments or data, or a new treatment of old data sets. So leading to, but the original discussion, there should be some additional of information. So that is, as a rule of thumb, at least 30% of the content must be original. So authors submitting such work are required to seek permission for reuse of the published conference paper if the author does not hold the copyright. Like if the conference proceedings or whatever the published material, if the author holds the copyright, then he can use it, but he has to add more original content and cite the conference in the acknowledgement section or the references section if applicable. And now most of the journals, they are also providing this repositories, submitting the manuscripts early in the repositories. So these preprint services also applicable to frontiers. Like we can submit the preprints. Yeah, so you can submit any manuscript as a preprint. But once if it is submitted to frontiers, and then if the files are being shared to any preprint repository such as Archive or PRJ or OSF, then it should not contain the frontiers logo or branding, because it is still not published in Frontiers journal. But before that, we are sharing to any preprint repository, so it should not contain any frontiers logo. And the one more important thing is conflict of interest statement. This statement is very important whenever we are submitting any kind of manuscript to any journals, especially frontiers. So a conflict of interest can be anything potentially interfering with or could be perceived as interfering with a full and objective peer review, decision making, or publication of articles submitted to the frontiers. Like any kind of things like personal, financial, or professional affiliations or relationship, this all can be perceived as conflict of interest. So all these things must be mentioned clearly in the statement of conflict of interest. Similarly, this both the authors as well as members of editorial boards, like editors or reviewers, all should comply with this conflict of interest. So now we'll see about the research ethics. Like all research submitted to Frontiers for consideration must have been conducted in accordance with the Frontiers guidelines on study ethics. So in accordance with the scope guidelines, so Frontiers reserves the right to reject any manuscript that editors believe does not uphold high ethics standards, even if authors have obtained ethical approval or if ethical approval is not required. Example, if any author has performed a study and he has done ethical clearance, but if Frontier checks that manuscript is not upholding high ethical standards, then it can also be subjected for rejection. And similarly, Frontiers encourages authors to follow the ARRIVE guidelines for design, analysis, and reporting of scientific research. So these ARRIVE guidelines. So whether the screen is visible regarding ARRIVE guidelines?
No, cannot see, Soma.
Okay. So I opened a link, hyperlink, but it's not showing. Anyway, I will just explain. So these ARRIVE guidelines, they state 10 essential points for any kind of studies, like study design, sample size, inclusion and exclusion criteria, randomization, blinding or masking, outcome measures, statistical methods, experimental animals, experimental procedures, results. So these all 10 essential points that each item of these guidelines includes examples of good reporting from the published literature extracted from different types of studies in model organisms ranging from mammals to invertebrates. So all these things, studies should follow this clear 10 sets of guidelines which are given by ARRIVE. So, but I cannot show this. So anyway, so this ARRIVE guidelines, this is more important, which is followed by Frontiers regarding the design, analysis, and reporting of scientific research. And if we come to specifically to the Frontiers in pharmacology, so there is a guideline called the 4 pillars of ethnopharmacology. So every manuscript should follow the requirements based on these 4 pillars. So that is under 4 categories, like pharmacological requirements, requirements specific to the composition of the preparation, basic experimental and ethical requirements, article type specific requirements. Like, Just a minute, I will close and open that. Now, can you see the arrive guidelines?
Yeah. Yeah. This is ethic for, yeah.
So this ethics, whenever we are doing any animal studies, so what are the things which should be considered for studies on animals? Like for ethical standards related to animal experiments. So is ethical approval and a written informed consent from the owners of the animals needed? And if so, does the manuscript contain the appropriate statement, and has any necessary ethics approval been issued by appropriate committee or organization? This is important. And if the manuscript uses animal samples, does the manuscript clearly state where these samples were obtained? Does the manuscript mention that experiments were performed according to the named location or national guidelines, like Committee for Purpose of Animal Experiments, like CPCSA guidelines, which are followed in India? Similarly, national guidelines, whether they are followed for any kind of animal experiments, and whether they follow the internationally accepted standards, and what are the whether it contains a clear description of the procedures performed on animal subjects, including any details of any anesthesia or euthanasia performed. All these things must be mentioned in the manuscript. Like there are some checklists. So mandatory checks include ethics committee approval and full name of the ethics committee provided for all vertebrates and higher vertebrates. So this study, if it is conducted on animals, this statement must be given with full name of the ethics committee. If this ethical committee approval, if not required, then a statement specifying that, along with justification why this ethics approval was not obtained, or why the ethics committee waived the requirement for approval. This statement is also very important. So all procedures performed on animals described in the manuscript appear to conform to internationally accepted standards and euthanasia method, as mentioned earlier. And additional checks include the dose of anesthesia provided and conforms to approved standards. Animal or organ cells, if they are used, their origin is whether clearly mentioned or not. If zebrafish is used, then that is also required ethics clearance, except for embryos and larvae up to 5 days old. So this is one example I'm mentioning, but this total list, this is available in the Frontiers website, journal website. So every author should check before preparing the manuscript for Frontiers. And as I mentioned, these 4 pillars of best practice in ethnopharmacology. That is, first point is the pharmacological requirements. So ethnopharmacology, what we deal with the ethnomedical information. So based on use in traditional use of any kind of natural products or whatever may be. So those things we have to develop data based on recent scientific techniques. So the traditional context must be described in the introduction and supported with bibliography, primary references. Why we have chosen the particular product or particular traditional medicine for further studies. And this basis, we have to, there should be a clear testable research question, like example hypothesis. But we might be knowing in our traditional medicine system, there are some terms, they are explaining about a particular illness, but those things which cannot be studied using advanced techniques. So those kind of things like, I'm not getting the exact word. Okay, so credible experimental models. So the methods must be state of the art and all are a credible alternate resulting in a better understanding of potential pharmacological effects. Like example, if we do antioxidant assay, simply publishing antioxidant assay using this FRAP, ABTS or DPPH methods, they are not acceptable. So these are generally chemical assays, but to get a chemical profile of a preparation, Only with these things, we cannot expect a particular formulation provide good antioxidant effect in clinical setup or preclinical setup. And similarly, any studies reporting the antimicrobial property, if you perform the disk diffusion experiments, it should be followed by in vitro or in vivo experiments. And the specificity must be assessed to rule out general toxic effects, because the antimicrobial effect may be due to the generalized or overall toxicity of the chemical or the molecule. So that's why we have to rule out general toxic effects, like doing cytotoxicity tests parallelly. And also the mechanism of action must be assessed in sufficient detail. Without that, this data cannot be published. Similarly, inflammation, anti-inflammatory studies only with the kind of rat animal model. As we know, the rat hind paw edema model, that is not acceptable unless it is a part of a larger pharmacological or phytochemical studies. Similarly, in silico studies like molecular docking studies, only with computational approach, we cannot publish. So it should be followed by any bench work to confirm the affinity. So if we are performing drug interaction studies or drug protein binding studies, it should be followed by either in vitro or in vivo experiments to provide or to give some experimental pharmacology information. We all know that recently this network pharmacology is also being considered for many traditional medicine system, because many traditional medicines, they have multiple components, and they are targeting multiple different proteins or disease targets, so that this network pharmacology can make a map out of a treatment approach, so that these kind of studies must critically assess the pharmacological evidence to evaluate the potential effects of a preparation or herbal product, the limitations of the evidence. So all these things, other points also there, we have to consider while submitting any in silico network pharmacology studies. And one more important thing is single dose studies are mostly not acceptable unless if it is a novel or new compound which is not yet studied in detail, and we have to provide justification on ethical grounds. So most of the studies and with ethnopharmacology or traditional medicine systems, that should be done in multiple dose basis. So this is related to the pharmacological requirements. Composition requirements. As we know, these natural products are mostly isolated or extracted from plants or marine or many other different sources. Uniformity, content uniformity is the most important thing. And from where this source, we are sourcing this particular raw material, that is one more important thing. Because the same plant which is collected from a particular region in one country may not be similar to another plant, the same plant which is available in another country. So that whenever a manuscript is published using a particular extract from a particular country, it cannot be reproducible in another country. So that is why the most important thing is what the name of the plant with the species and all complete taxonomy details must be provided. And if you are preparing some extract, then all the procedure, how it is done, everything should be clearly described, like solvent protocol, drug solvent ratio, everything to be provided. And whatever the extracts or fractions or whatever if we are studying, so those things it should be published along with HPLC or UPLC chromatograms or to prove these are the particular compounds present, not like the groups of compounds like categories, alkaloids, glycosides, not like that. It should provide compounds, identification of specific compounds using HPLC, UPLC, or even HPTLC chromatogram may also be considered. So if we are using commercial extracts, then if batch number is mentioned, then that is fine. We can use the data from other published literature also if batch number is same, because that is the same product. So if it is a purchased natural product, then purity, the supplier name must be included, because anyone wants to repeat the similar kind of studies, then they will also follow the same thing. And for extracted compounds, we have to perform the purity and how it is done, the quality tests which are performed, all these things must be clearly stated.
due to time,
Yeah.
Soma, can you make it faster? Because Prof.
Yes.
Wawai Muli will add something.
Yeah, sure. Okay, thank you. So the last one is article type specific requirements like field studies or reviews, systematic reviews, meta-analysis, and all these things, they have specific requirements. So if we go through the requirements of 4 pillars of best practice in ethnopharmacology, we can get more detailed information. So that's all from my side. Thank you.
Thank you, Soma, for the explanation of the lecture. I heard before that Prof. Wawai Muli will going to add something or
yes.
explain more.
Yes.
Yeah, please, Prof.
Yeah, okay. Thank you very much, Dr. Soma Sondaran. So actually, Mr. Soma Sondaran is my junior when we are studying in Gata University of Pharmacy. So, okay. I would like to add something, about my experience as an associate editor of Frontiers in Pharmacology. Just on the 4th slide. Yeah, can you able to see? Yeah.
Yes.
Okay. So I myself, as an associate editor, I should be registered in a Loop. Loop is a kind of web page. This is, originated from foreign frontiers. So all the author and the reviewer, so it should be registered via Loop. So this is my Loop profile. So I myself, I am an associate editor for the Frontiers of Pharmacology. But the Frontiers of Pharmacology have many sections. One of the section is about experimental pharmacology and drug discovery. So because of my major is basic pharmacology, so I will only edit the manuscript based on like a research study. This is pharmacology, not edit for like clinical study. So I also mentioned that the Frontiers of Pharmacology have many sections. One of section is experimental pharmacology. The other is about ethnopharmacology. Dr. Soma already mentioned about the ethnopharmacology section. So if you interest to submit like phytochemical study, so you have to do some requirement like phytochemical analysis, quantification analysis, not only screening, but you have to provide HPLC data, for example, GCMS, LCMS data like that. So as an associate editorial, I already, I just 1 year joined with the Frontiers, but this is for you information, this is unpaid job, unpaid worker, yeah, this is voluntary job. So as an editorial already 1 year, so I already edit 6 manuscripts and then review for about 4 manuscripts, and I also can be joined as a research topic. Okay. So this is 2 research topic that we create. So I create 2 kind of research topic. This is anti-inflammatory agents with Dr. Soma Sondaran and another colleague from Japan. So in the research topic, we can invite some contributor author from our colleague to submit the manuscript related to this topic. So recently I also create a research topic related to the method and application of experimental pharmacology with the other colleague from the other country. So as an associate editor to publish an editorial, so it is no need to pay anything without article processing charge. So regarding the article processing charge, there is some issue that Frontiers have a high APC. But for your information, we can get any discount. So in my experience, with my student, so we can get discount up to 50% or 40%, 60% like that. So you can submit the form for discount APC in the website itself. Next, I would like to share you some article that edited by me. So majority is from Asian country, from China, and then from Saudi Arabia, and also from India. So Frontiers not only accept the original article, but they also accept the methodology. So like this example, an in vitro platform to study reversible endothelial to mesenchymal transition, it is kind of methodology article. This is the other three of manuscript that I already edited. Okay. And then finally, I would like to inform you about the reviewer process. So as an associate editor, I responsible to invite the reviewer. So actually, similarly with Nature, Frontiers also have data or what to say, database for a potential reviewer based on the expertise. I mean, they will select by the keyword like that. But in one article, one manuscript that I handle, so I invite my colleague, my Indonesian colleague. So I invite Flori Ratnasari. Flori Ratnasari is also high reputable researchers from Indonesia. And then at the time, so she's very I mean, interest to review this manuscript. And then I also invite one from Faculty of Pharmacy, University Gadjah Mada. So it means the reviewer process in Frontiers is very strict, So because of I know who is the reviewer. So Flori Ratnasari and Retno Murwadi is expert in this kind of field. So I mean, there are some issue that the review process in Frontiers is not good. I think we can ignore it. So I think that's all from me to add some information related to my experience as associate editor of Frontiers of Pharmacology. Thank you very much, Dr. Agus.
Thank you, Dr. Somasundaram and Prof. Muli for the explanation about I think the Frontiers also had the strict guidelines, strict requirement with, of course, with the high ethical concern about should be fulfilled by the authors. Now we going to ask Dr. Nafrialdi as a panelist to give a comment or maybe further question for both speakers. For Dr. Nafrialdi,
Thank you.
please.
Good afternoon, Dr. Agus Rizal, Dr. Roy Muli, and Dr. Somasundaram. And also thank you for the committee for inviting me to participate in this discussion. Maybe I'm not was the question as the right author, but maybe since I was also experienced as the editor in another journal, so I like to share our experience in managing the manuscript and managing the publication of our journal. As the associate editor, do you also do the initial selection of the manuscript? Because I have experienced that very lot of number of manuscripts that come to us as a chief editors. How about you as the associate editor? Do you also select first which one to be proceed, which one to be rejected the first time? What do you do, Dr. Soma or Prof. Muli?
Yeah, thank you. Actually, we receive like a lot of manuscripts for either we have about our comments. Based on, we will just go through based on the abstract or whatever the figures, whether the quality of the figures and other things. And also this, whatever the guidelines I mentioned about the ethics, what they follow. So these are the basic some aspects, and they do preliminary check actually. So the preliminary evaluation is done by the system itself. It will give the comments and suggestions, like these are the possible things where you can look into. Like there may be the statement which is mentioned, the ethics statement, it's not clear. So the manuscript contains some human information, patients, but they have not mentioned the statement about any human ethics committee clearance. So then we will get some idea where to go and check whether this is done as per ethical guidelines or not. Similarly, animals are mentioned in the manuscript, but there is no statement regarding the animal ethics committee. So like that, the system itself will give suggestions for various aspects like plagiarism and other things. Similarly, during every point of review, we'll get information regarding if the reviewer has given some comments and the number of words in the reviewer comments is very less. So whether you accept this giving proper review of the manuscript or not, like that, we will get artificial intelligence based results, so that will help us in deciding which part we have to look into. And initially, if we think that this manuscript is not suitable, then there are 4 different things, like as per ethics, or it's not meeting the journal requirements or standards, like that. Then we can give the comment to the chief editor. Then he will also go through, and whether he can directly reject it, or he will, once again, he can send it to another reviewer or another associate editor comments. So that can also be possible.
Okay. So if you compare with the previous presentation from the Nature, I have impression that all manuscript come first to the chief editor, and then the chief editor divide it into send it to the sub editor, maybe, or associate editor. What is done in Frontier, Prof. Muni?
Yeah, similarly, I mean, because of we also have another activity, so I myself, I only can accept, for example, 1 year only 2 or 3 maximum. Yeah, this is the first thing. So I will make sure whether I have time to review, to edit it or not. So this is the first things I have to consider. And then, yeah, so the chief editor, there is a preliminary check, yeah. And then after it is complete, it will come to associate editor. And then after that, we can through it from abstract and everything, yeah, as Dr. Soma already mentioned. And after that, we can make a decision whether it is rejected or go through the review process. So, and then I would like to add some information. The decision whether it is rejected or accepted, not give by me as associate editor. So I just only give the recommendation. So after completed the review process, then everything will go to the chief of editor. So you know that the review process in Frontier is very open, yeah. So everyone can see the process, like reviewer 1, reviewer 2, the interaction, yeah. We call it interactive reviewer. Is it true, yeah, Dr. Soma? Yeah. So they think that it is everything is open. So no need to hide something. Thank you.
That initial review is done blind. That initially whatever the process is happening, an initial review by the associate editor, and it is sending to the reviewer, everything is done blind. But once after getting the review comments from the reviewers, then we will assess the comments. Then if everything is okay, or if the manuscript is having some quality or some not having much issues, then we will open the interactive session. So that interactive session where all the authors can see what other reviewers has given comments, and there itself they will respond. Again, the reviewer can ask another question, like it is a continuous process. Still, he endorsed the manuscript. So the reviewer, once after all these corrections, he will endorse the manuscript. If both or minimum 2 reviewers, if they endorse, then we can, editor or associate editor, we can also give some comments. If something is there, then the chief editor will accept it. We can recommend either for acceptance or revision, chief editor will take the final decision.
Okay. Is there still any time for me, Dr. Agus?
Yes, we still have a time.
Okay. Another question. I find when we submit a manuscript, then the reviewer make an argument or some question which should be followed exactly, precisely. Yeah. My question is, as the associate editor, do you also add some comment which is you consider is not include in the question of the reviewer, but you consider it maybe is important, or you just rely on the two reviewers? This is the first question. And the second question is, I find that frequently the journal has the very precise manuscript format. Yeah, why the style of writing, style of figure and table, etc. But when there is something is not appropriate to the style of in-house style of the journal, they send back to the author. My question is, what is done? What is done by the editor then? Because almost everything is done properly by the author, but almost nothing corrected by the editor after that. Okay, that's my question. Thank you.
Yes, so I would like to answer the first question from Dr. Nafriadi related to what is the role of editor. Yeah, so I mean for the reviewer process, we give opportunity to the reviewer. Yeah, I mean 2 or more reviewer. So after the reviewer process is complete, so I also should make any opinion, any recommendation, any suggestion for the authors. So, I mean, in the interactive review process, we have, I mean, as an editor, we have like a box to submit some comment, suggestion, or something to the authors themselves. So, this is for me. Yeah, I always make any suggestion, even though the reviewer process already complete. But I also want to make any suggestion to improve the quality of the manuscript. So maybe Dr. Soma will add something.
Yeah, the same thing. Even the reviewer comments, they accept or reject. Even there are possibilities that if we invite 3 reviewers, 1 may reject. But we should have minimum 2 reviewers acceptance, or what we can say, the number should be more number of reviewers. If they give acceptance, then that can be considered. But meantime, both the chief editor as well as associate editor all have access to this interactive review. So everyone can give their own comments and suggestions. For those also, the authors have to respond. Then only articles can be endorsed or accepted. But I didn't get your second question. Yes, I think you are mentioning about the journal requirements like tables or figures, why they need in specific format. That's what your question? Or?
Yeah. I mean, if the author submit which not very precisely according to the requirement of the journal, do you make correction yourself, or you send it back to the author to make it appropriate according to the journal style?
No, actually, the figures and tables, all these things, the journal, they can't do any changes. It should be, as such, it should be published. Because if something happens during this process, they are not responsible for that. So that's why they're asking everything should be submitted in the required format. Like editing the page, that is different, the wordings. That also, final approval is from the author side. Everything is open here in Frontiers. Everything is, you can see as such. And the figures or tables, as per requirement, if you are providing, then they can be published as such. Because it is online version only. So you can clearly see how it fits in the journal.
Okay. Thank you, Dr. Agus Rizal.
Okay, thank you. I think there is 1 or 2 last question that I saw it. The first one is, I heard there is a presentation that Frontiers also accept the preprint article. because preprint article is already had the DOI. So how is the regulation? I think based on your both experience about to accept the preprints article. And the second one is actually the last question is how many, based on your experience, how many % that you reject the article that sent by the chief of editor to both of you?
Yeah. So the first question, the preprints, now we all know the preprints is like the total manuscript is as a repository. It can be published, but the copyright will belong to the author only. So that he can publish it in any journal, not like in a repository. So after publishing, we have to provide a link in the preprint version or in the repository that will take you to the journal publication, the final publication site. That is what Frontier says. Page also should have a link to access to the original site.
Okay.
And the second question is,
how many % that you reject?
Almost I can say that rarely, maybe 2 or 3 articles I rejected. Apart from that, more than 80% got acceptance, because there are every chance to get corrected in the review process. There are many, even there are some journals, some articles, they took 1 year to revise again and again. Not like many journals earlier, only one revision or second revision, they will give a recommendation for rejection if it is not corrected. But in Frontiers, you have many different time points, like you can get extension to revise your manuscript, and finally it will be accepted. Very rarely it gets rejected, because there is no limitation for number of articles in an issue or something like that.
Okay.
So it's very different
Thank you
from
very much
medical
for
journal, Dr. Rizal.
Yeah.
I think in medical journal Indonesia we reject a lot of paper.
Yeah, but it's, I mean, every journal have.
Sorry for adding, because of there is a preliminary check. So because of the manuscript that already sent to associate editor, it is through the preliminary check, like a specific requirement. So that's why I think the quality of the manuscript is good. Yeah. So the problem of rejection, in my case, because of they cannot approve kind of raw data. So I already 2 manuscripts rejected by myself because of they cannot provide raw data. So they, as the reviewers asked about the raw data, but they didn't send it. So that's why, and then they make a reason that we don't have that specific data, so we will withdraw this manuscript like that. Yeah. So I mean, the important things is as the author.
Yeah, here one more thing, like one manuscript got rejected.
Yeah, as an author, we can get an extension. So for example, we got 2 weeks to revise it, but we can make an extension. So during the extension, so we can do for example additional experiments, something like that. That's all.
Yeah, one more thing, like one article got rejected based on they have used 5 grams of a dose in animal study. So that was because they have done everything, all scientifically proved everything, but the dose which is not matching the clinical use. So that is a major reason it got rejected. So this is one important thing that is also mentioned in the guidelines also regarding the dose selection for the study.
Okay. There is a nice
thing, Dr. Agus. Yeah. So about the raw data, it is very important because of majority Frontiers journal asks for raw data, including also the statistical analysis. So I mean, everyone, who interested to submit to Frontiers, please keep your raw data in Excel, for example. So everything should be kept in the Excel data, including also the statistical analysis. Figure everything like histopathology analysis, something like that.
Yeah, I'm really sorry. I mean, now it's getting I mean, there is hot discussion at the end of the time, but we are very sorry that we have to close the session. Thank you very much, Dr. Sundaram and Prof. Muli, and of course our panelist, Dr. Nafrialdi. So it is interesting. So maybe I'm a little bit give us a resume that Frontiers Journal actually is already showing that I already mentioned they have a strict guideline requirements and also support high ethicals issue. So I think there is no doubt that about this journal. And interestingly, actually, we can learn as an author that the Frontiers have how to say it, as mentioned as Dr. Sundaram, that we can get extension when we wants to revise our manuscript. But of course, again, that Prof. Muli mentioned about the raw data now. I think there are more journals asking about the raw data. So thank you very much for all, and also the participants who still joined this session. I close the session now. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh. I give back to the MC for the next event.
Thank you.
Thank you very much to Dr. Soma Sundaram Arumugam, Professor Wawai Mulia Aroza, Dr. Nafrialdi, and Dr. Agus Rizal Ardi for the insightful session towards the end of our event. Ladies and gentlemen, we have now arrived at the end of CMARS 2023 Day 1. We hope every single person in the participants had an amazing experience today, and most importantly, learned plenty from the lectures given today. Don't forget that CMARS 2023 still has 3 days, which will be just as exciting as today. Be sure to join again tomorrow for Day 2, where there will be more amazing speakers and lecturers. We would also like to remind every participant of this webinar regarding the attendance form that you can find on the chat box in the Zoom. The attendance for that we will also send through your email along with the feedback form. Please kindly fill out the form directly afterwards this event so CMARS can continue to improve and provide satisfaction for every participant. This event would also not be successful without our sponsors, Novartis, Novo, Prodia, AstraZeneca, and Widya Genomic. Thank you to all sponsors and media partners who have supported CMARS 2023. As an appreciation to all participants, we would again like to invite all of the participants, speakers, and committee to turn on your camera to take a group photo. Please, you can turn on your camera. As we wait, I will give the floor to the documentation team to take the photo.
Okay, thank you, Natasha. 1, 2, and 3. And next slide, 1, 2, and 3. And the last slide, 1, 2, and 3. Thank you. I will give back to the MC.
Thank you very much to the documentation team, and thank you for those of you who have turned on your cameras. Lastly, we would like to express our gratitude to all of the participants while also apologizing for any inconveniences during this event. Once again, thank you for your enthusiasm for this symposium, and see you tomorrow for CMARS 2023 Day 2. Thank you very much. You may now leave the meeting.